Hello, everybody. Welcome back to day 4. So in day 4 here, what we're going to do in this first hour is finish molecular biology, and then after that, we're going to get into genetics for 3 hours tonight and most of tomorrow, and then we'll finish genetics tomorrow. So now a lot of stuff coming up your way, a lot of modern molecular biology in this first hour. And now let me say is, for some of you, this might be conceptually difficult stuff, and but you got to be paying attention. I know tonight is the NFL draft. I know a lot of you are going to be distracted. You're going to be watching ESPN or something to see who drafted number 1 or whatever. Don't get distracted. Don't get distracted tonight. Yeah, I'm dying to know who the Steelers drafted 15. But I'm not going to be distracted. I'm not going to say, hey, let me go look to see. I'm not going to, so don't get distracted with all this other things going on. I know the NBA is on tonight, looking to scores, because if you lose sight, if you get distracted with some of these things, you could be missing some very important points. And then you can come back to the computer and say, what did Turco talk about? Well, it's because you were distracted. So let's, so we'll be in good shape here. Okay, as long as you're paying attention. So in chapter 7, the last chapter in molecular biology, what this is about is techniques of genetic analysis. So we talked yesterday, finished off yesterday, spending a lot of time about the RFLP. Now the RFLP, restriction fragment length polymorphisms technique, that technique is it's used, it's a highly used, often used for DNA analysis. And the basis of the technique is the southern blot. Okay, and we talked about the southern blot and how the probe determines everything in a southern blot. Okay, and then, which again is the basis, the laboratory basis of the RFLP. So now what's coming up next is, so again, at the end of the chapter, you're going to see an RFLP vignette coming your way. So we'll practice this. We're also going to practice RFLPs in genetics. I apologize for coughing, but over the last couple of days, I've been under the weather. Man, I know I got a touch of the flu. My temperature's up a little bit, and little achy, all this kind of stuff like that. But hey, I can deal with it. I can handle it. You know, grow up. You know, I gotta grow up. You know, quit whining about things. So I'm here. So don't worry. So if I cough, I apologize. But it's just it's the condition I'm in right now. But I'm glad to be here. I'm excited about being here. And my adrenaline will keep me going here because I want to be here. Okay, so now so we talked about the southern blot, and now what's coming up next is the northern blot. And so we'll talk about what the northern blot is used for, how it can be used, and then we'll finish off talking about another technique in genetic analysis, and that's the PCR reaction. Okay, so the first thing is the northern blot. Okay, now the northern blot is, what it's about is, what does a northern blot measure? What a northern blot measures is, it measures, let me see your answers here without looking in the book. What does a northern blot measure? What's it used to detect? That's right, it's used to detect RNA levels. So what we're looking at here is a northern blot of the expression of the FMR1 gene in this example. So, so there's a particular gene. It can be made into RNA, and the northern blot is going to be used to analyze that RNA. Now, let me ask you this: another question here, and that is, what disease is the FMR1 gene involved in? What disease and pathology is the FMR1 gene involved in? That's right, it's Fragile X. Okay, but again, this northern blot on page 105 has nothing to do with Fragile X, nothing to do with the pathology. It's simply just analyzing a particular gene, and it just so happens it's FMR1, but it has nothing to do with Fragile X. Okay, now we'll talk about Fragile X later on. It's a very high yield disease for boards. We'll talk about it later on in genetics. Okay, now what I'm going to do here is I'm going to give you a quick 2-sentence oral vignette. Pay attention, because I'm going to give you a 2-sentence quick oral vignette, and then I'm going to ask you a series of 5 questions that's based on that simple vignette. Okay, ready? Here comes. Here comes a simple oral vignette. Suppose they say to you that messenger RNA was taken from skeletal muscle, brain, liver, testes, lungs, pancreas, and heart. After the messenger RNA was taken from the cytoplasm of those tissues, the RNA was subjected to electrophoresis and then analyzed using a probe that detects the FMR1 message. There it is. That's the quick oral vignette. Okay, everybody, so everybody understand that. Here comes the first of 5 questions by looking at this northern blot. Now, the first couple questions are very, very simple. Okay, now, so the first one will be either a yes or no question. By looking at the northern blot, can you say, is the gene expressed in the brain? You got to know what the verb expressed means, right? Is a gene expressed in the brain? We talked about that in chapter 1, as well as in chapter 5. So is a gene expressed in the brain? That's right. And the answer is yes. What that means is that in the lane of the brain, there are many messenger RNAs. Many messenger RNAs in this lane in the brain that are different sizes. And one of the messenger RNAs is the messenger RNA for FMR1 that's detected by the probe. Question number 2. Again, a simple question. By looking at the northern blot, yes or no, can you say, is the gene expressed in the liver? Yes or no? Is the gene expressed in the liver? And the answer is no. What that means is again, if I take messenger RNA from the cytoplasm of the liver and subject the RNAs to electrophoresis, in this lane right here of the liver, there are all kinds of messenger RNAs that you cannot see with a naked eye. There's messenger RNAs for all the blood clotting factors, messenger RNAs for transferrin, messenger RNA for albumin, for ceruloplasmin, for ALCAT enzyme, and so on. So in the lane of the liver, there are many messenger RNAs that you cannot see. And then when it's probed, using the probe that sees the FMR1 message, nothing is seen. So what that says is the FMR1 message is not present in the liver. Simple question and simple answer. Question number 3, slightly harder, just ever so slightly harder. Okay, now how would you explain this? Why in the lungs it's a thin band and in the brain it's a very thick band? Why the darker signal in the brain compared to the lung? Let me see your answers here. That's right. Yeah, well done. That's right. So what it means is that the brain expresses more of the messenger RNA than does the lung. The lung makes a little bit of the messenger RNA, the brain makes a heck of a lot more, and the more that's present, the darker the signal is when it's probed, using the probe, in this example here, for FMR1 message. And so the thickness or the intensity of the band, the darkness of the band, it actually is giving you a rough estimate of how much might be present. Matter of fact, the northern blot could be used to get a rough assessment of HIV infections. So if somebody's an HIV infected, because HIV is an RNA, if you electrophoresis the RNA of the virus on electrophoresis, if it's a thin amount of it versus a thick amount of it on a gel, it's telling you one person is going to be very highly infected with the virus. If it's present but only in a very light band, they're mildly affected. So again, the intensity of the band gives you a rough idea of how much might be present. Question number 4. Now, before you respond using the public chat pod, think about your answer first. Here comes the question. By looking at the northern blot, can you say, is the gene present in the liver? Can you say, is the gene present in the liver? And the answer is no. You cannot say that the gene is present in the liver. And why? Because I know some of you are saying, well, if the gene is present in the brain, it's got to be present in the liver. Okay, and that may be true. That may be true, but I never said in the vignette that the tissue was taken from the same cadaver, same person. I never said that the RNA was taken from the same person. So now, if it did say from the same person, in other words, if they said a cadaver was taken and the RNA was taken from each of the following tissues of the same cadaver, if the gene is present in the brain, it's got to be present in the liver, because in that person, it's in all the cells if it's present in one tissue. So, but otherwise, if you want to know if the gene is present, you got to do a southern blot. Even 2 different people, you cannot say yes. You cannot say yes if it's 2 different people. Why is that? Because there are examples. In one person, they have a gene, and another person, they don't have the gene. Can you think in pathology? What's the best example of this? In pathology, where somebody has a gene, and another person, they are missing that gene. The gene is deleted. What's an example in pathology of that? Let me see your answers. That's right, Uruj, that's right. Daniel, that's right. Prader-Willi is an example of a disease in which there is a gene deletion. That's coming up a little bit later in chapter 1 in genetics later on this evening. So the idea is in Prader-Willi, that's an example where one person has a gene and another person who has Prader-Willi is missing that gene. So again, now Mikala, you said CF. CF, it's the basis of the mutation. It's a 3-base deletion of a codon. The gene is still present. The gene for the chloride channel protein, CFTR, that gene is still present in cystic fibrosis. But there's a mutation within the gene. It's a 3-base deletion. Not the gene is deleted, a codon of the gene is deleted in cystic fibrosis. Okay, is that true, Michaela? Do you understand that? Okay, good. Okay, so again, so what I'm getting at here is be careful when you get a vignette about northern blots and so on, or if they give you various tissues. You got to know, read it very carefully if it's coming from the same individual. Otherwise, you got to do a southern blot to see if the gene is present. Now, the 4th and last question is the hardest one. You can imagine, this is often on the national board, you can imagine that on board exams, they might have an arrow pointing to the heart. Here, pointing to the heart. And they might ask the why question. Here it comes, get ready with your fingers. Why is that RNA in the heart smaller in size because it migrates faster than the other tissues? Why is it smaller in the heart compared to the other tissues? Everybody chance that's right. Now, there's no protein here. So now there's no protein. This is RNA. This is not protein. There's no protein here. Different size palindromes. There's no palindromes in RNA. Remember, RNA is never fragmented. RNA is never cut up. So again, it's not has nothing to do with palindromes because this is not DNA. This is RNA, and RNA is never fragmented with endonucleases, and this is not protein. And so the answer is no. I see a lot of you got the question correct, and that is that's due to alternative splicing. Now remember yesterday when we talked about alternative splicing, I said to you it's very high yield on board exams. They love the topic of alternative splicing. But and then we talked about yesterday several ways they can ask you about alternative splicing. The idea, for example, of well, I guess it was the day before that, but we talked about the idea that certain a lot of different proteins in immunology as well as in endocrinology, the dopamine receptors are generated by alternative splicing techniques in a cell. But so they can ask you about alternative splicing by that means. But this is another way they can ask you about alternative splicing so that you understand what that means. Is that what it says is the spliceosome in the heart splices the RNA for FMR1 differently than the spliceosome in the brain, testes, and lungs. And this is all normal stuff. It's all normal. It's just the spliceosome in the heart splices it differently in the brain. Now, don't worry about the mechanism. Well, how does it know to do that in the heart? Don't worry about that. Just know what it means. Just know what it means that the heart is splicing it differently than in the brain. Now, some people would think, well, why is it smaller in the heart? Maybe because of a mutation or whatever. No, it's not a mutation either, because if it was mutated in the heart, it would be mutated in the brain of the same individual. So the only way you can have a smaller size is the normal situation of alternative splicing here. So please be ready for a northern blot. I hear about northern blots and RNA electrophoresis on boards quite frequently, especially the topic of alternative splicing. High yield stuff. Okay, now what's coming up next is again, if you have questions, please use the Q&A pod because Dr. Seda and Dr. Donald, they're excited to start giving, they want to help you out and give you answers to your questions. So please use the Q&A pod if you have questions here. All right, now what's coming up next is the last technique that's discussed in this chapter. In genetics, I will be talking about another technique called the dot blot for genetic analysis. But in this chapter, the last technique we're going to be talking about is the PCR reaction. PCR. Now, what does it stand for? It stands for polymerase chain reaction. Now, what this is about is it's probably the single most powerful technique in molecular biology. It's a very powerful technique. What this is about is this is where the PCR is where what it means is simple as I can state. What that means is you're amplifying a piece of double-stranded DNA. You're taking a little bit of a double-stranded DNA, most often a gene, and making millions and millions of copies of the exact size of that double-stranded DNA. Okay, so again, the concept is PCR. You're taking a gene, for example, and by PCR reaction, you're making millions and millions of copies of the exact size of that double-stranded DNA gene. Now, you know where you first heard about the PCR reaction? It's a relatively new technique, maybe the last 12 or 13 years, something like that. Where you probably first heard about it was in the movie. There was a movie called, great movie, and the movie was called Jurassic Park. Now, if you watch that movie and you listen very carefully in the movie, what happened was there was a number of scientists. There were scientists in the movie that what they did was they took a mosquito that was trapped in amber in the dinosaur age. What happened was 150 million years ago, some mosquito bit a dinosaur to get some blood. And so when the mosquito is now sucking up the blood of the dinosaur, in that blood of the dinosaur are white cells. And white cells have the dinosaur's DNA. So then the mosquito takes the blood, and just so happened that the mosquito is then trapped in amber. 150 million years later, the amber is removed, and in the movie, the scientists then dissect the gut of the mosquito. Then they take the DNA of the dinosaur, and if you listen very carefully, they say we are using PCR, polymerase chain reaction, to amplify the dinosaur's DNA. And as a result, they ended up with Brontosaurus and Tyrannosaurus Rex and raptors and all these type of things. It was a great movie. You know why it was a great movie? It didn't star Brad Pitt. That's why it was a great movie, because he would have trashed the movie. But it was a great movie, and in fact, there was a lot of science in that movie. Anyway, that's what the PCR reaction is about. We probably first heard about it. But nowadays, we use PCR in reality not to amplify dinosaurs. Although you might have heard that in Siberia there's I read about this in some magazine or whatever. And in Siberia, some mammoth was trapped in ice, and no, they're taking and some scientists are taking the mammoth's DNA, and several years from now, because there's going to be a lot of problems, they're going to try to by using PCR to try to come up with a living mammoth. We'll see if that happens, but anyway, the woolly mammoth, that's right. But in any case, in reality though, in your profession, I mean, what do you use PCR for? And it's obviously not used to make more animals. What it's used for is for genetic analysis of disease or other things. Now, to use the PCR reaction, now in your book, there is a diagram that shows the methodology of the PCR reaction. I'm going to skip over that. I don't want you to get bogged down in how you do PCR. If you want to know about it, it's in your book. You don't have to know that details the methodology of the PCR reaction. How many cycles you have to do a PCR, and when do you get double-stranded DNA of the exact size. Don't worry about that. All you need to know is to do the PCR reaction, you need a special DNA polymerase. Don't worry about the name. It's a special DNA polymerase. And because DNA is going to be made, we need all four deoxynucleotide triphosphates. We need deoxy ATP, deoxy CTP, deoxy TTP, deoxy, which one am I missing? GTP, all in it. So we put them in a test tube, add DNA polymerase along with that DNA. So we're going to amplify a piece of the DNA. And this is the key to understanding the PCR reaction. Remember earlier in the RFLP reaction that uses the southern blot, the basis, the most important part of the southern blot is the probe. In the PCR reaction, the basis, the most important thing, the key for you to understand, it's the primers. Remember, DNA polymerase requires primers to make more DNA. And so we always need, when you hear the word PCR, that's telling you that there are two sets of primers that are being used. One for the 3' end of each strand of double-stranded DNA that we're going to amplify. So we always need two sets of primers here. And now let me stress here, in a test tube, the primers are DNA primers. In the body, the primers are RNA primers. But DNA polymerase doesn't care. As long as it sees a primer, it's going to make more DNA. So again, one more time. In the body, DNA polymerases use RNA primers, but in a test tube, if I take some of my DNA and I put it in a test tube and I want to amplify a piece of DNA, I want to amplify a particular gene, all I do is I add DNA polymerase and then I add DNA primers that are going to be specific for that particular gene. So now where this is being used, again, it's widely used. The PCR reaction is widely used. As an example, we're not going to go into the detail here, but as an example this is the best technique So on board exams, if they ask you, which of the following is the best technique to do this or that, if you don't know the answer, and one of the options is PCR, put it down if you don't know the answer, because it's likely to be what they're asking you about. PCR is the best method for genetic analysis. But as an example, here is HIV infections. So as you know, there are many ways, not many, but there are different ways to analyze HIV infections. For example, in immunology, you can use the ELISA assay. But the problem with the ELISA assay, there's a lot of cross-reactivity, a lot of false positives. A better assay for HIV infections is the Western blot. It's a very powerful technique, is the Western blot. But even more powerful is the PCR reaction. So the idea is how does this work? So what's done is, suppose I want to examine my white cells to see if I'm infected with HIV. So what I do is I take some of my white cells and put them in a test tube. Theoretically, all you need is one cell. Theoretically, all you need is one cell. And so to do PCR reactions. So I take some of my white cells, put them in a test tube, get rid of all the proteins with detergents. I got negative DNA in that test tube. And then in the test tube, I'm going to amplify one of the genes of the HIV virion. Now, it's not like you're going to amplify the entire virus. You don't have to. But one of the genes of the virus, for example, the GAG gene. In my body right now, I should not have the GAG gene. The GAG gene is in the retrovirus. It's not normally in me unless I'm infected with HIV. So now that I know, if I want to test to see whether I'm infected with HIV, what I do is I go to the computer and I say, tell me the sequence of the GAG gene. And within seconds on this monitor in front of me will come the sequence of the GAG gene. Remember, it's going to be one strand. It's the coding strand. The 5' end is on the left. Now that I know the sequence of the GAG gene, then I call a company, or I could do that in the laboratory myself. I make primers on the 3' end of both strands of the GAG gene. The primers are commercially available. You can go to Walmart probably and buy the primers for certain genes or whatever. I don't know. But the idea is so the primer is not a big deal. You can commercially purchase them or make them on your own. But the primers are designed to base pair with the 3' ends on the opposite strands of the DNA. So then the primers are present. We add DNA polymerase along with the nucleotide triphosphates, and DNA will be made, double-stranded DNA of the exact size that you're interested in. All right. Now, with that in mind, that's all you need to know. Don't worry about the methodology. If you want to know, it's in the book, but you're wasting time. So now the issue is how, besides HIV, like for example, so if I'm being tested for HIV, if take some of my white cells, put them in a test tube, add the primers for the GAG gene, and then you add DNA polymerase, and then you come back the next day. If in that test tube there's millions of copies of the GAG gene, that means my wife's going to say, "Where have you been?" And but if if there is no GAG gene present in that test tube that's being amplified in millions of amounts, then I'm safe for HIV. I didn't get infected with HIV. So there it is. That's the concept here. And so why it's a powerful technique, because theoretically all you need is one cell, theoretically, to do this. All right, now but the technique here, the PCR reaction, I'm going to, instead of doing using PCR to analyze a disease, what we're going to do here is talk about the use of the PCR reaction to do. Oh, and by the way, so the primers will find why they're also so important. Besides the fact they're needed by DNA polymerase, they find the DNA sequence that you want to amplify. So it's like, so if I'm infected with HIV, in my chromosomes, there's the GAG gene. It's like trying to find a needle in a haystack. That one gene in like 46 chromosomes, how are you going to find it? A needle in the haystacks. The primers will find it. The primers will look at all the chromosomes, look in the base pair. If the GAG gene is present, the primers will base pair. You amplify the GAG gene in millions amounts there. So again, the primers are the key to the PCR reaction. Probes are the key to the southern blot. Please keep that in mind. I'm emphasizing over and over again. All right, now what we're going to do here is talk about the use of the PCR reaction as a basis of another technique called genetic fingerprinting. Just like the southern blot is the basis of a technique called RFLP, PCR reactions are the basis of another technique called genetic fingerprinting. And genetic fingerprinting, just like RFLP, can be used for person identification, can be used for other reasons like genetic testing of diseases and so on. But the genetic fingerprinting is widely used in forensics and crime scenes. Somebody's DNA is being amplified, analyzed to see who is the perpetrator of a crime. You can do genetic fingerprinting technique, or you can do RFLP technique to analyze that DNA. But here, we're going to talk about genetic fingerprinting for a specific reason, and that's paternity testing here. You always hear about this, right? In recent years, if you're in the United States, you hear about this a lot. Some baby is born, and they want to know who is the biological father of that child. Matter of fact, a couple of years ago here in the United States, there was a widely known example of this. What happened was a few years ago in the United States, there was a very rich lady, and she had a child, and she died of a drug overdose. And what happened was, and all of these men came out of the woodwork to say, "Hey, I'm the father of that child. It's me. All these men came out of the woodwork and say, "Hey, I'm the father of that child." Why? Because they cared about the child. It had nothing to do with the money that the child's going to inherit. But the idea is that they. By the way, who was that woman again? What was that? Jessica Simpson. No, what's her name? Very rich. What's her name? Oh yeah, that's right. Anna Nicole Smith. That's right. Anna Nicole Smith. Very rich lady. Had that child, and she died. And then all these guys came out of the woodwork and said, "Hey, it's me. I'm the father," because they cared about the child. It had nothing to do with the money. Nothing to do. And something wrong with my eye. Okay. Anyway. So but they figured out who was the biological father. And how they do that? The technique coming up right in front of you here. And that is the genetic fingerprinting technique. Now, what this is about is, here is what's being analyzed, what's being amplified by PCR. And what's being amplified is related to something we talked about yesterday. Now, remember yesterday we talked about, if I took your chromosomal DNA and mine and your neighbor's DNA, your DNA sequences are going to be different from me, different from your neighbor, and different from everybody else in this world. So the DNA sequences will be 99.9% identical, but there will be a little bit of DNA that will not be identical, and that gives rise to the variation. And then yesterday we talked about why is your DNA not identical to mine, and one reason of two reasons. One reason is due to mutations. A lot of times you have mutations in spacer DNA that will not cause pathology. Remember the SNPs? We talked about the SNPs. But if a mutation is in a gene, that gives rise to pathology. And now the second reason that we didn't talk about yesterday, we're going to talk about now. The second reason why your DNA in sequence is different from mine is due to something called short tandem repeat polymorphisms, abbreviated STRPs, or sometimes you might see it called STRs, all different names for the same thing. Short tandem repeat polymorphisms. This is where pieces of DNA can be amplified. Regions of DNA can be amplified. Now, when these regions of DNA can be amplified, it depends how big it is, what you call it, what you call that piece of DNA. And when the piece of DNA is amplified, generally it's called VNTRs, variable number of tandem repeats. Variable, listen to the words, VNTRs, variable number of tandem repeats. So VNTRs is the class of these repeats that can be amplified. But when they're very small, when these VNTRs are very small, 2 to 6 nucleotides in length. So when they're very small, these repeats amplified many times over, they're called STIRPs. So again, STIRP is one kind of a VNTR. When the regions that are when the DNA that's amplified is very small in length here. And now these STIRPs are sometimes called microsatellites. Microsatellites when they're very small in size. When they're larger, you might see them called mini satellites. When they're very large, you might see them called satellites. But these are all called VNTRs. It's a class, and one sub class of it is the STIRPs. And it's the STIRPs that are widely analyzed in genetic fingerprinting for paternity testing. Now, what do you mean by a short tandem repeat? Like what? For example, you could have 3 nucleotides that's amplified one after another, after another, after another. Now you might say, well, won't that cause pathology? If you have a repeat one after another, yes, that would cause pathology if it occurs in a gene. And if it occurs in a gene, if you have a STIRP occurring in a gene, it gives rise to Huntington's disease, Fragile X, myotonic dystrophy, spinal bulbar muscular atrophy, or Friedreich's ataxia. Remember all those 5 triplet expansion repeat diseases? We have the codon, like for example, in Huntington's disease, it's the codon CAG, 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 one after another, after another, after another. That's a STIRP. It's a STIRP. 3 nucleotides. It's a STIRP that's amplified multiple times, one after another, after another, after another. But that's in a gene causing pathology. But you and I can, for normal individuals, we can have STIRPs in spacer DNA, and it causes absolutely no pathology. You and me have STIRPs in our chromosomes in different regions in spacer DNA, causing no pathology here. So again, don't be concerned that these STIRPs are causing diseases, and the only time you do is that if it's occurring in a gene. Just like mutations. Mutations, if it occurs in a gene, gives rise to pathology. If you have mutations of DNA in spacer DNA, no pathology. Same thing here with STIRPs. If it occurs in a gene, pathology. If it occurs in spacer DNA, no pathology. So now the concept here is, suppose, in this example, suppose we have a child. Here we have a child, and here is the mother of that child, and here's some male that's now going to be tested to see if the male is the biological father of that child. So what's done is to take DNA from all these people. Now, how do you take the DNA? How do you take the DNA? Yeah, it's very simple. All you need to do is get a little swab, a Q-tip. Watch, take a little swab, back of the cheeks, and take that swab with the cheek cells and put it in a test tube. All you need is one cell, one epithelial cell from the cheek. No invasion, no biopsy, none of that kind of stuff. And just all you need is a swab of the cheek cells, put it in a test tube, and you got that person's DNA in that test tube. That's how simple this technique is. So in that test tube, then we got DNA from the mother. We take another test tube, take a swab from the child, and another test tube. We take a swab from the cheek cells of the tested male. Okay, now in the mother's, in the test tube that has the mother's chromosomes, in reality, there is a particular STIRP that's located on chromosome 7 that's often used in paternity testing or genetic fingerprinting. On chromosome 7, there is a STIRP called GATA. Now suppose what you're looking at here, this is one of the 2 chromosomes that the mother has of chromosome 7. Remember, the mother has 2 chromosome 7s. One came from her mother and one came from her father. And somewhere in that in chromosome 7, remember the human chromosomes, the human genome, human chromosomes have been sequenced. We know where that GATA STRP is located. It's located on chromosome 7, and suppose it's amplified 6 times in a row in the mother, in one of the 2 chromosomes that she got here. So she has 2 chromosome 7s, and in one of her chromosome 7, she has the GATA amplified 6 times in a row. GATA, 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 6 times in a row. Now suppose she inherited this particular chromosome from her mother. Okay, so she inherited that chromosome 7 from her mother, and that STRP is present 6 times that she got from her mother. Now also, remember, what you're looking at here is one of the 2 strands of that chromosome 7. The other strand is not shown. It's implicit. But please remember, there are 2 of these strands of chromosome 7, of that particular chromosome. All right, now what about the other chromosome that the mother inherited from her father? So that same GATA is, let's say it's amplified 10 times in a row. So the chromosome 7 that the mother inherited from her father, that GATA is amplified 10 times in a row. GATA, 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 right in a row, one after another, 10 times in a row. Okay, so that is now the genotype of the STRP. In one chromosome it's 6, and the other it's 10 times in a row. You and I, if I took your chromosome 7s, both of your chromosome 7s, you have a GATA that might be amplified 5 times in one chromosome, 12 times in the other. In me, I never tested it, but that GATA in my chromosome 7 might be amplified 6 times, and the other chromosome might be amplified 10 times, something along that line. So again, this is a STRP that's often used in reality. Okay, so this is the genotype now of the STRP that the mother has. Now, what about the tested male? So the tested male also has 2 chromosome 7s. And suppose in a tested male, one of his chromosome 7s has a GATA STRP amplified 6 times in a row, not causing no pathology because it's not in a gene. It's in spacer DNA. So that STRP is amplified 6 times in a row. And suppose in the other chromosome that the tested male has, that STRP is amplified 15 times in a row. So all in the chromosome 7 that tested male has. Okay, so we take DNA from the mother, the child, and the tested male and put them into 3 separate test tubes. Okay, and now then what happens is, so then in each of the 3 test tubes, we then add primers. Again, primers are the key to PCR reactions. So because we know the sequence of chromosome 7, we know where that GATA STRP is located, and we know what's the DNA sequence on the flanking sides. On the 3' end of the STRP, we know what that sequence is because we know the sequence of chromosome 7. So then I can go to Target, I can go to Walmart or whatever, and say, hey, I want you to make me primers, 2 primers for the 3' ends of that particular STRP. So the primers will come the next day in excess amounts. I'm adding the primers to all 3 test tubes, and the primers now will look at the chromosomes, look in the base pair, and they will base pair as shown by the arrows on the 3' ends of that particular STRP. So these arrows are representing both primers. Each primer base pairs with a 3' end of each strand of chromosome 7. Now, if you're not following me here, if you're thinking, man, what's the 3' end? That was stuff we talked about on Monday. You know, the idea of primers and 3' ends and all those type of things. But the idea is we need 2 primers that'll base pair with a 3' end of this STRP. Okay. So we had primers in the mother's test tube. We had primers to the tested male's test tube, and they'll base pair with a chromosome 7, and we had primers to the test tube that has the child's DNA, and the primers will base pair with that STRP on the flanking sides of the STRP. Okay, so then what's done is you do the PCR reaction. You come back the next day. How simple it is. You come back the next day. You take the amplified DNA, and what's the amplified DNA? The amplified DNA is the STRP, GATA, because you're making millions of copies of double-stranded DNA of the exact, I cannot emphasize enough, it's the exact size of DNA that you're looking for. All right, so then you take the amplified DNA and then subject it to electrophoresis. Now suppose in electrophoresis, by calibrating the gel, suppose the 15 mer bands here, the 10 mer bands there, mer meaning polymer or whatever, the 15 mer bands here, the 10 mer bands there, and the 6 mer of the GATA bands there. How do we know that? We just calibrate the gel. We know that the 15, 10, and the 6 band at these various places just by calibration. Okay, so now what comes up next is simple. How do you interpret the PCR gel here? How do you interpret? Because the GATA is being amplified by PCR, the amplified DNA is subjected to electrophoresis, and how do you interpret the results? And you always see two bands. Always see two bands. Why? Because one of those bands was in a chromosome that was inherited from that person's mother, and the other band was coming from a chromosome that was inherited from that person's father. So always two bands. All right. Now the way you interpret the PCR gel is very simple. Very simple. All you got to do is look in the child first. And in the child, you say which of the two bands in the child was the band that was inherited from the mother. The other two bands in the child in case one, it's the lower band in the child that was inherited from the mother, because the mother has the band, the child has the band, and the child's inheriting that band in the mother. Now the child has it. And then you go to the other band in the child, and that other band in the child had to be coming from the child's biological father. And in this example, case one, notice that the tested male also has that band that the child has. So let me see your answers here. So what is the conclusion? What's the conclusion? So the child has the mother's band, the child has the tested male's band. What's the conclusion of the paternity of the child? No, 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 no. No, no, you cannot say he is the father. All you can say is he could be. He could be the father of that child. Okay, so stay tuned. So again, one more time. If you do this technique and the tested male has that band that the child has, your conclusion is the tested male could be the biological father of that child. Now let's go to the case two. Now in case two, again, you look at the child first and say which of the two bands in the child was the band inherited from the mother. And clearly it's the lower band. The lower band in the child was inherited from the mother. And then we go to the other band in the child and ask, did the tested male have that band? And the answer is no. Now what's your conclusion about case two? Let me see your answer. What's your conclusion about case two? That's right. A little bit of hanky-panky took place. A little bit of side action, a little bit of hanky-panky with that mother business. Ooh, oh, that's right. Because the tested male cannot be the biological father of that child. Because that band in the child, the upper band in the child, had to be coming from that child's biological father. So we can quickly eliminate that tested male as the father of that child. Now coming back to case one here. So if we get case one where now the tested male does have the band that the child has, then you can say that the father could be that the male could be the biological father of that child. So now how do we get rid of the verb could be to the verb is the father of that child? How do we replace the verb could be the father to the verb is the father? Two different things. How would you do it? How would you do it? Just think. That's right, Sagra. There we go. That's right. That's right, Bobby. That's right. You just to do. Let me go back. I pressed the wrong button. To have a definitive identification, then about 8 to 10 different stirps on different chromosomes would have to be analyzed. And that's what that in reality is what's usually done. 8 to 10 different stirps on different chromosomes are analyzed, and if in each case the tested male does have the band that the child has, at that point there, statistically, there is no doubt that is the biological father of that child. So the idea is if you just do one stirp, it's could be, but it doesn't have to be. But if you do more and more and more stirps, the more stirps you do, statistically, then there's no doubt that is the biological father of that child. So again, that is the concept here. So again, let me conclude this. So paternity testing using the PCR reaction, it's very simple. Again, don't worry, don't get bogged down in the minutia. Don't get bogged down in the details. How you do the PCR, just understand what it means, and go through it, because it's very simple technique is the PCR reaction. Okay, so now if Steve can now bring up the selection pod, because we're gonna. I'm looking at the hour, the time here. It's 10 minutes to the hour. So what we're gonna do is we're gonna do 2 quick questions here. Then we're gonna finish off molecular biology. So we're gonna do 2 questions, and then we'll take a 10-minute break. Okay, so I'll give you 1 minute per question, and these 2 questions are not easy. Let me stress right now, they're not easy. And the first question, it comes from your book at the end of the chapter. I just changed some words around, but it's related to a question in your book. The second question is a question that I wrote. Okay, so here is the first question. Let me move this out of the way here. Okay, so it's not an easy question, but here is the clinical vignette. Here is the first question, based on stuff we talked about. I don't know, Steve, where we can move this. Okay, maybe. Oop. Can't. It doesn't sit there. Okay. Okay, there we go. Okay, Roy. Okay. Roy has it. Can you see the question? I see people are saying you can't see the question. Maybe we'll move the selection pod over the public chat pod. Okay, there we go. Maybe try it that way. Okay. I'll give you another 20 seconds here. Ten seconds. Okay, so let's close the poll, end the poll, and broadcast the results. Roy, if you could broadcast the results so all the students can see. Okay, here we go. Stop it. Oh, this out of the way. Too many pods. That's the problem. Right, there we go. So everybody can see. Okay, here we go. Thank you. Too many pods. By the way, the students out there, you may not know this, but in front of your screen, you just see a couple of rectangles in the screen. You see the camera or whatever. But me, and as well as the other people that are running the show here, it's much more complicated. It's all these different pods. There's a list of the participants, the students, attendee list, Q&A stuff, all presenter chat pod. It's like being in a cockpit in an airplane. So many things you gotta look at, and this and that. It's much more complicated. And that's why sometimes I get distracted, because I'm looking over here, looking over there, and but anyway, it's more complicated. Okay, it looks like so 2/3 of the students put option B. And let me move this out of the way. And the answer to this question is indeed option B. All right, now before we talk about this, What they're talking, why, before we explain why option B is the answer, let's look at the vignette. They're saying in the vignette that this boy, parents of a boy with CF, they talk to a genetic counselor, the mother's pregnant and wants to know about the fetus. Again, on board exams, that is a typical board question. They'll show you a pedigree, they'll have a diamond, and that's usually the fetus, and they want to know what is the outcome of the fetus. And so in genetics, in the next hour, we'll get into issues of Punnett squares and all these type of things, and probability of having disease or being a carrier. But here, the power of the RFLP will tell you exactly what is the outcome of the fetus regarding cystic fibrosis. All right, now they're saying that we know the mutation is in exon 10. So there are a lot of exons in the CFTR gene, and the mutation is in exon 10. So that region of exon 10 is going to be analyzed by RFLP. So the idea is, the concept is, suppose this represents in yellow exon 10 of a very long chromosome that goes in both directions. There's spacer DNA, and here's exon 11. The rest of the exons of the CFTR, the rest of the chromosome, not even shown there. But what they're saying here is, in this vignette, is then suppose in the vicinity of exon 10, where the mutation is, suppose there are palindromes for a particular endonuclease. Notice here, they're not telling you what the endonuclease is. All they're saying is that DNA, the gene is being cut by a specific endonuclease, generating fragments, which are being analyzed by RFLP, by Southern blot. So the idea here is, in the vicinity of exon 10, as shown by the purple vertical lines, suppose that represents a palindrome. So in the region of exon 10, you're going to get a fragment that's going to have this size right here. See, from that purple line to that purple line, you're going to fragment. And that's what's being analyzed in this Southern blot here. And notice there are no sizes. There's no numbers like kilobases, none of that kind of stuff, no KB, none of that kind of stuff. You don't need that information. You don't need the sizes per se. It's the relative sizes. Notice how there's some large bands and there's some small bands. And they all have that mutation. So now the question becomes, how is it that it's the same fragment, yet sometimes the band is large and sometimes the band is smaller in size? And the answer is, it's due to those palindromes we just talked about in the last figure. Suppose in the vicinity of exon 10, in spacer DNA, as shown by that orange rectangle, that represents some stirp. Whatever that stirp is, it doesn't matter. It could be AT or something like that. But there's a stirp right near that exon 10 within the palindrome, within the fragment. And suppose that stirp is present one copy in a particular individual, and in another individual that stirp is amplified 2 times. It might be amplified 3 times in a row. Again, that's not causing pathology. It is simply a stirp in spacer DNA that can be amplified in a person multiple times. But notice, it's going to change the length, the size of the fragment, because how many stirps is present will change the size of the fragment. Okay, now on the left side over here, now coming back to this, how are we going to get the answer of the fetus? Now, let me ask you this. You should know this without me telling you this. Cystic fibrosis. What is the mode of inheritance of CF? What is it? What is the mode of inheritance of CF? Every medical student should know this. That's right, it's autosomal recessive. Now, in autosomal recessive, again, we'll talk about it in genetics in the next hour. In autosomal recessive diseases, capital A is the normal allele, and little a is the mutant allele in autosomal recessive diseases. I'm going to color code it with purple being capital A normal, and blue is the little a, it is the mutant gene, mutant allele. Okay, so what I'm going to do now is notice, and now notice, when you go and look at the pedigree, You know the father and the mother are heterozygous carriers because they don't have pathology. You don't see solid circles or solid squares. So the mother and the father are phenotypically normal, but they are carriers, both of them, because the son has the disease. And because it's autosomal recessive, the son must have both genes that are mutated. And then we're going to analyze the fetus. So now, again, as I mentioned yesterday, you must always go to the person you know most about genetically first. Now, if you went to the father, because the father is heterozygous, if you went to the father first, one of these two bands in the father is going to be capital A, and one of these two bands in the father is going to be little a. So if you went to the father first, you would not know which of the two bands is capital A versus little a. Same thing with the mother. One of these two bands is going to be capital A, one of these two bands is going to be little a, but you don't know initially. You always go to the person you know most about genetically. And who do you know most about genetically? Obviously is the son. Now in the son, let me ask you this. Get ready with the responses. The upper band in the son, is that capital A or is it little a? The upper band in the son, is it capital A or is it little a? That's right, it is little a. The lower band in the son, is it capital A or is it little a? That's right, another little a. Now, how'd you know that? Because the son is homozygous with mutant genes, causing the pathology that the pedigree says. Okay, and in fact, the vignette says a boy with cystic fibrosis. So the son has little a, little a. So now, again, don't get bogged down. Why is it that the band is of different sizes, yet it's still little a? Because it depends on how many stirps are present in that fragment. How many stirps were present in that fragment determines the size. And one of these bands was inherited from the mother, and one of them came from the father. All right, now let's go to this son. In the son here, let's look at the lower band first. The lower band, was that band in the son, was it inherited from the father or was it inherited from the mother? Where did it come from? That's right, it comes from the father. So why the mother doesn't have that band? The mother does not have the lower band. The father does. So therefore, the lower band in the son has to be inherited from the father. So we can make now the assignment little a in the father to do the inheritance. So the son gets that gene coming from the father. That's the lower band. Meaning the upper band in the father got to be capital A, because the father is heterozygous. The pedigree says so. The father is heterozygous, so the upper band got to be capital A, normal gene. Now this band, the upper band in the son, has to be coming from the mother, because the father does not have that band. The upper band has to be coming from the mother. So now we can make the assignment. The upper band in the upper band in the son is now the lower band in the mother, and that's got to be little a, because the son is inheriting this allele from the mother. And the mother, again, is heterozygous. That means the upper band in the mother got to be capital A. Notice how we're doing this. We start with the person we know most about genetically, and then start backing off and say, where did the bands come from? How were they inherited? And starts to, it's like solving a puzzle. You ever work with puzzles and so on? The hardest part of a puzzle, with all these pieces, the hardest part of a puzzle is starting with a piece that you know most about, and usually some corner, the corner piece. And once you figure out that corner piece, then things fall into play. Oh yeah, this comes the next thing. Things fall into play. I mean, me personally, I like solving RFLPs because it's like solving a puzzle here. All right, so now that we know the father and the mother's genotypes here and bands, now let's go to the fetus. Now, if we go to the fetus, the upper band in the fetus, notice that the father has that upper band, and so does the mother. So initially, we cannot make the assignment. Was the upper band in the fetus, was it coming from the father or was it coming from the mother? We don't know initially. So therefore, we go to the lower band in the fetus. Notice the father doesn't have it, the mother does. So therefore, the lower band The lower band in the fetus has to be coming from the mother, so we can make the assignment. That's going to be little a, coming from the mother, and therefore to do the inheritance. And therefore, the upper band got to be capital A to do the inheritance. And so, therefore, what is the answer? The fetus has inherited the mutation from the mother, but not the father. That's why the answer is B. Okay? All right, so I know it took a long time to go through this question, but we had a lot of explaining to do. All right, now let me just say here, right now it's 3 minutes after the hour. We're going to take a 10-minute break right now. I decided let's take a 10-minute break now because the next question, I want you to be fresh. Next question is a typical board question. It's one of these triple jump questions. You have to figure out something to figure out something else to figure out the answer. And I want to make sure you're fresh. I don't want to blow it by you. So let's take a 10-minute break, and then we'll go back to this vignettes. Okay, so I'm going to go off camera. See you shortly. Okay, everybody. Let me say something about this question. There's going to be other questions in genetics. The last chapter in genetics also have very similar RFLP type questions. And it might have looked, I was looking at the public chat pod. I noticed a lot of you were saying, hey, well, I went too fast, or how can you get this done in 1 minute? What took a long time was explaining the issue why the bands are different sizes and the issue of the stirp. This is a type of question that they're not going to, this information here about the stirp and the palindrome, you don't need that information. And you just have to get this answer, all you got to do is look at the pedigree and say, who do you know most about genetically? And you know it's the son. Make the assignments little a, and then you say, well then, where'd they come from? And you can get this question answered in less than a minute. Not because you're a pro at it or I'm a pro at it or something. No, it's a matter you have to go to the heart of the answer. You notice there's no numbers here, no molecular weights, no issues of the endonuclease, but this is a typical board question. Typical board question. You can't sit there and dawdle and think about other things that are going on in your life when you're taking the board exams. You got to focus, and get to the heart of the question and get to the answer. This is it looks. I know for some of you it's difficult because history tells me so. It's difficult for some of you because you never had this before, or been a long time ago, and you're these southern blots and but you have to practice. You have to practice. At the end of the chapter, there's some more practice questions, other questions that you can practice on. There's no substitute for practicing. Once you practice this a few times, things start to fall into place. But the key is you always start with a person you know most about genetically. Just try me out. Just try it out. You got to practice here. You must practice. Okay. All right. Now, what's coming up next is another typical board question. But again, as I mentioned, it's a triple jump question. These are the hardest types of the USMLE questions. These triple jumps, where they give you information, you have to figure out something. And then based on what you figured out, you figure out something else. And then after you figured out that, then you get to the answer they're looking for. All right? 1 minute. I know on board exams they give you 78 seconds. But well, I'm giving you only 60 seconds. Why? I'm trying to speed you up. I'm trying to get you to think, focus. You got to practice that. You got to practice focus, and you got to practice working a little faster. You can't dawdle here. And that's what I'm trying to get you to do, to try to think quicker on your toes, quick here. All right, here comes the next question. Then we're done with molecular biology. Here it comes. Ten seconds. All right, and we'll stop the poll and broadcast the results. All right, and looks like the majority of students put option D, and the answer to this question is option C. Okay, now, yeah, Natalie, by the way, when you said, yeah, that's right, don't complicate things. Yeah, I noticed what you wrote, Natalie, and that was very astute. That's why I try to keep things simple for you. I try to simplify as best as I can. And sometimes you have to explain something, but I try to keep it simple. Don't worry about the details here. And don't overcomplicate issues, but try to keep it simple. Now, this question, every time I do this question for medical students, most students put option D, and the answer is option C. Most people think the southern blot, but it is the northern blot. Now, this question is, again, a typical board question in that they talk about some disease and they're going to ask you what's the best technique to analyze that disease. And again, on board exams, at least one question will be on that type of technique that's going to be used for analyzing a particular disease. All right, now, in this vignette, let me ask you this. Get ready with your fingers. What is the name of the disease this girl has? What is the name of the disease? That's right. Not sickle cell. No, it is beta thalassemia. That's right. She has beta thalassemia. Now, how do you know this? Now remember earlier I mentioned to you that on board exams, always pay attention to the first phrase. Because sometimes, not all times, but sometimes in the first phrase, they give you a clue. You always pay attention to the age of the individual. Always pay attention, male or female, because it could be X-linked disease or something like that. Always pay attention of the ethnic group or religious group or geographical group. And here is a geographical issue. They're saying an Italian girl. So again, what that means to you and me, well, if it's an Italian, that must be likely they're asking me about some disease that is in the Mediterranean countries. And so what diseases in the Mediterranean countries primarily? You would say G6PD deficiency, and you would say beta thalassemia. So it could be those. But you know it's not G6PD deficiency. Let me ask you this. What is the biggest clue in the first phrase that tells you it's not G6PD deficiency? What's telling you? For me, I'm in there you go. Well done. See that, Faiza? That's right. That's right. Lewis, that's right. But look how beautiful. That's right. Daniel, look how nice that was. That's right. Because that one word, girl, is telling you it's not X-linked recessive. I mean, it's theoretically possible, but highly unlikely an X-linked recessive disease will be found in a female. It's possible, but highly unlikely. So again, this clue is they're telling you, don't think G6PD deficiency, think the other disease that's found in the Mediterranean countries, and that is beta thalassemia. Now, what confirms it as beta thalassemia? And one is the hemoglobin levels are very low. Another major clue is the histological examination of the peripheral blood smear. They have target cells. Now, you know what a target cell is in pathology. It's those red cells that have a bullet right in the middle of the red cell. And another characteristic of beta thalassemia is the acanthocytes. And what a acanthocyte is, it's those red cells that have these spikes on them. And so the spikes are these acanthocytes. By the way, sometimes a student will ask me, well, why aren't all the cells either target cells or acanthocytes? I mean, why? Look how many cells are normal. Look how many red blood cells are normal. It's only a few of these are target cells and acanthocytes. And why is that? And the reason for that is because if all the cells were acanthocytes, you'd be dead. You couldn't carry oxygen anymore. You got to have a lot of red blood cells that are normal that are carrying oxygen around for me and you, as we'll talk about next week in metabolism. But so it's not that all the cells are abnormal. Just some of the cells are abnormal. If all of them were abnormal, yeah, you'd be dead. All right, so now here is this. So again, that's the characteristic of, that's telling you this is beta thalassemia. That's the first jump, is you have to figure out this girl has beta thalassemia. But they're not asking you what's the name of the disease. Here comes the second jump. They're saying, which of the following techniques is most likely to show the most common cause of this condition? So here's the second jump. What is the most common cause of beta thalassemia? Let me see your answer. What's the most common cause of beta thalassemia? Most common cause. the biochemical cause, the defect. What's the mutation? That's right. Well, Kashif, good try. I forgot, but on board exams, they don't want to. I forgot will not be an option. It's not going to be option E. I forgot. It ain't going to be. You got to know it. You got to know it. That's right. And excuses. That's right. That's right. And the answer is the most common cause. It's a splicing defect. It's a splicing defect. Remember that beta. We talked about this the other day. That beta globin has 3 exons and 2 introns. And in beta thalassemia, there's a mutation, a missense mutation in the splice donor site of intron 1. And so therefore the spliceosome does not recognize it as the splice donor site. Therefore intron 1 is not removed. And if intron 1 is not removed in beta thalassemia patients, and it is removed in normal patients, that means the RNA, that the messenger RNA in beta thalassemia patients is going to be larger in size compared to normal beta globin messenger RNA. So the idea is, which of the following techniques, now here comes the third jump. The second jump is knowing what is the cause of the disease, and now the third jump, based on your information to figure that out, what technique analyzes RNA? And the answer is the northern blot. There it is, the northern blot, because it detects abnormally sized RNA, different sizes of the RNA. There it is. It's not overly difficult, not complicated, Natalie. That's right. Don't make it complicated. That's right. It's just a matter of thinking. Just, okay, I know this, I know that, and then get to the answer. Now let's go through the other options and why they're incorrect. Now, option A, we haven't talked about the dot blot yet. We will tomorrow. But the dot blot, it cannot distinguish differences in sizes of the RNA. Remember, you probably forgot this, but at the beginning of the chapter, when we talked about the various names of the blots, and I said the dot blot, the big difference between a dot blot and the other ones like southern, western, and northern is there's no electrophoresis in a dot blot. No electrophoresis. No separation on the basis of size. So a dot blot would not be useful. In the LISA assay, it tests for the presence of an antibody or antigen, not RNA. This is an RNA problem. Option D. Now, a lot of students put option D. And the reason, and now the southern blot would likely not detect the mutation causing the defective splicing. Now, what I mean by this is, to explain this. That's why it takes time for this question. You have to do a lot of explaining. The idea is, suppose this represents the beta globin gene in some chromosome. And here, remember, it has 3 exons in the orange and 2 introns in the yellow. And the mutation is right here at the splice donor site of intron 1. So that's what's defective. And that's why you cannot remove intron 1. That's why the RNA is larger in size in the disease. Now in a southern blot, remember the DNA is cut up. And suppose that, to analyze that fragment that has the mutation, you were to cut the DNA here, as shown by the purple here, and the purple line there, you get a fragment size that would not be changed. The length of the fragment is not going to be different in normal versus beta thalassemia patients. Because if it's a missense mutation, yes, there's a 1 nucleotide change in the splice donor site, but the length of the fragment is not changed. That's why a southern blot is likely not to be useful. The only time it might be useful is that if where the mutation is, there's a palindrome, like you saw yesterday with sickle cell anemia. In sickle cell anemia, that mutation changed the palindrome. But here, the mutation is not changing the palindrome. So please understand the concepts. Not complicated, it's just a matter of understanding. Now the Western blot is, we detect the presence of a protein, but not whether it was defective. So Western blot might be useful, but the best technique on board exams, they often say what is the best technique or most likely to determine the outcome or something like that. So other techniques might be useful or whatever, but usually they're going to say what is the best technique. Okay, there it is. All right, so now we're done with molecular. And by the way, again, this is a typical board question. It brings in biochemistry with molecular biology, obviously with pathology here. So you gotta know how to do these questions here. Okay, now what's coming up? And so now we're done with molecular biology, and now we're going to get into towards the end of the book is we're going to get into medical genetics, which is the last 6 chapters of the book. So this is section 2 and chapter 1 of medical genetics. Now let me stress here, in genetics, there are some very high yield chapters, like chapter 1, extremely high yield, and there's some very low yield chapters, and I'll point those out once we get to them. As long as you're paying attention, as long as you're not being distracted by the NFL draft, as long as you're not being distracted by the NBA games tonight, as long as you're not distracted. If you go away for a minute, I might say this is low yield, and you didn't hear those words, and you'd be spending an hour or two. What did he talk? Oh, and I would have said low yield. Don't worry about it. Pay attention. All right. All right. Now this is a high yield chapter. Out of 322 questions, you're going to see, in my prediction, you're going to see at least one pedigree on the board exams, at least one mode of inheritance, which chapter 1 is all about. So you've got to be able to analyze pedigrees, modes of inheritance. Now, before we talk about the 5 modes of inheritance, there's some terminologies that I want to make sure, definitions that I want to make sure we understand. Here, I'm not going to go through all these definitions, but just to make sure, so there's no, I don't want to leave any stone unturned here. But the gene, again, we talk about genes in molecular biology. A gene is a basic unit of inheritance, a piece of DNA. But I want to stress, because sometimes students it never dawned on them. So a gene is made into RNA, but the RNA doesn't have to be messenger RNA. The RNA can be tRNA, and the DNA that encodes for it is called a gene. So a gene encodes for tRNAs. A gene encodes for ribosomal RNAs. So all 3 classes of RNAs are encoded by genes. So a lot of students never understood that issue. They think, oh, a gene encodes messenger RNA. Yes, that's true, but it also can encode for tRNA or ribosomal RNA. Later on, I'll show you why that's so important for you to know, that genes encode for the other 2 classes of RNA, not just messenger RNA. Now, the next definition is the locus. The locus is a location of a gene on a chromosome. Now, what I mean by this is, suppose we have 2 homologous chromosomes. The chromosome number is not important. It could be 2 versions of chromosome 1, 2 versions of chromosome 15, doesn't matter. But you have 2 versions of the same chromosome. One of these chromosomes came from your mother, and one of these chromosomes came from your father. And the sequences of these 2 chromosomes are not identical. They're almost identical, 99.9% almost identical, but not identical. And since they're not identical, you call them homologous chromosomes. They're homologous, but not identical. Now, somewhere in the chromosomes, there may be a gene. And suppose we have 2 versions of the same gene, and shown here in the orange. And where that gene is located, that's called the locus. So a locus is the location in a chromosome where a particular gene is located here. Okay, and now allele, we talked about alleles in molecular biology as well. And allele is different forms of the same gene. In other words, different DNA sequences of the same gene. If gene 1 is the identical sequence to the other gene in the other chromosome, then it's the same alleles, the same sequence. But one of the genes might get a mutation in it, coming from the ancestry of that particular parent. So if this gene here on chromosome B, somewhere in the ancestry of it, got a missense mutation, something like that, it's no longer the identical gene. And so we'll call it version 2. So these genes are called alleles of each other. They're not identical. It's the same gene, but they're not identical in sequence. So they're called alleles of each other. So the alleles could be identical, but they don't have to be identical. But they're referring to the same gene. One of them may have a different sequence. It might be one nucleotide change, something like that. That's called alleles. And depending on what, now genotype is the proteins encoded by that particular gene and alleles at this position. And so in other words, if gene 1 is the normal gene, then if you have 2 copies of it, your genotype would be 1, 1. If one of the genes has a mutation in it, then the genotype would be 1, 2. If both the genes have the same mutation, then the genotype would be 2, 2. So that's the genotype. It could be 1, 1, 1, 2, or 2, 2. That's the genotype of that locus at that particular chromosome. And depending on what the genotype is, it results in messenger RNA being made, and then that also gives rise to a protein, and one of the proteins might be defective if it's coming from the mutant gene. The other one's normal or something along that line, and that gives rise to the phenotype. So the phenotype is the physically observable features based on the genotype at this locus here. Okay, now the other definitions here, we'll get to those a little bit later. Okay, now coming up next is, so we're going to get into pedigrees. And before we get to the individual pedigrees, there are symbols that you find in pedigrees. And now I'm not going to go through this table. You can do this on your own. But one thing I'm going to call your attention to is just to make sure the most important symbols, every student should know this, is a square is a male, circle is a female, and a diamond is an unknown sex. On the boards, the diamond usually will refer to a fetus, but technically it doesn't have to be. A diamond could be this person of an unknown sex. So again, but boards, if you see a diamond, they're referring to a fetus. And in closed circles or squares, that is the phenotype showing pathology. They are affected. Now the carriers of a trait, they might show half open, half closed circles, half open, half closed squares. But they may or may not show you that. You saw that earlier in molecular biology. Remember the cystic fibrosis question? Remember the sickle cell anemia question, where both the mother and the father were heterozygous carriers, but phenotypically they were normal. So they don't have to show you who is a carrier of a particular trait. You might see an open circle, open square, yet they have a defective gene here. They don't have to show you that. Now the rest of these symbols, you can read them on your own. But in talking to the students over the years, what I hear about typical board questions in USMLEs, I'm certain with Canadian boards as well as COMLEX as well, typical board questions is regarding pedigrees. The first thing you always got to do is be able to recognize the mode of inheritance. Is this pedigree showing autosomal dominant? Is it showing X-linked dominant? That kind of stuff. You should be able to recognize the mode of inheritance. The second thing that I often hear about regarding pedigrees, you should be able to predict the recurrence risk. Now, what does that mean? That's the definition of recurrence risk coming up on the next figure. And then another thing I hear about, not as often, but sometimes I hear about this being on boards, they might show you a complicated pedigree, very complicated 3 or 4 generation pedigree. And they might ask in this complicated pedigree with multiple generations, who is an obligate carrier of a trait? What I mean by that is, in the pedigree, there might be people who are carriers, heterozygous carriers of a defective gene, yet they're phenotypically normal because they're carriers. Okay, and they're not showing you the half open, half closed circles and squares, but they might expect you. Who are those people? And so that's called obligate carriers. They must have a defective gene that's being passed on to the progeny. So they might ask you to identify obligate carriers of a particular trait. Don't worry, we're going to practice this. Don't worry, we're going to practice this in a particular pedigree coming up soon. We're going to go through it, and I'm going to ask you who are the obligate carriers of a trait here. So it's all coming up. Don't worry. Okay, now coming up first is the first of the 5 pedigrees, and that's autosomal dominant. Now with autosomal dominant, now the thing is, a lot of students may have different ways to figure out the mode of inheritance. It doesn't matter which way you use as long as you get the right answer, right? Whatever methodology you use, as long as you get the right answer is the most important thing. Now, what I'm going to do here is in the upper left-hand part, I'm going to tell you what I look for and to determine it's autosomal dominant. And if you don't like the way I do it, that's fine, as long as you get the right answer. But what I do is I look at a pedigree and I look to see, does it have a vertical appearance? In other words, you start at the bottom of the pedigree, work your way to the top, and notice how it has a vertical appearance, because the disease is in every generation. It has a vertical appearance, and the disease is in every generation. And it's characteristic of autosomal dominant here. That's right, Natalie, before you say it. Don't overcomplicate this. Make it simple. Just make it simple here. That's right. So autosomal dominant. Okay, now here comes that definition of recurrence risk. Now recurrence risk, the definition is, it's the probability that the offspring of a particular mating, that the offspring will express the phenotype, will show the disease, will show the pathology. What's the probability that when 2 people mate, they're going to have the disease, they're going to have the pathology, they're going to show the phenotype here? That's recurrence risk. Now there's another definition I want to make sure we attend to, and that is the definition of a Punnett square. Now a Punnett square, I think everybody knows this, but a Punnett square is a diagram used to predict the outcome of a particular crossing of a male and a female here. Okay, and these Punnett squares shown down here. Now in the Punnett squares, you're going to see these letters, and you must make sure these letters are straightened out, understanding what the letters are referring to. If you blow by these letters, capital A, little a, you're going to get hurt if you're not thinking here, because this will trick you up if you're not thinking. In autosomal dominant diseases, the capital A refers to the mutant gene, the mutant allele. It's dominant. It is a mutant gene, so that gene that's in it has some sort of mutation. That's the capital A. The little a is the normal gene here. Now again, please keep this in mind, because on the next figure will be autosomal recessive, and with autosomal recessive diseases, the letters are just the reverse. Okay, so again, the letters will really mess you up here. So capital A in autosomal dominant is the mutant gene, and little a is the normal gene. Now let's look at a particular cross, a mating. Suppose we look at this, a mating here of a male who has the disease and a female who's genetically unrelated. So the male is, because he has a disease, is capital A, mutant gene, little a. And the genetically unrelated female has to be, likely be little a, little a. Let me ask you this, just to make sure we understand this. Why is the male, oh, by the way, in these Punnett squares, the male typically is always at the top in a Punnett square, and the female's genotype is on the left side. So the male is at the top and the female is on the left side here. Okay, now in the male, capital A, little a, how do you know the male is not capital A, capital A? He could be having pathology. There are people with autosomal dominant diseases in which both genes are mutated. But how do you know he's capital A, little a in this particular pedigree? Let me see your answer here. That's right. Well done, everybody. Because his mother right here, his mother does not have the disease. And because it's autosomal dominant, that means he is inheriting the mutant gene from his father. His mother is giving him a normal gene because the mother's genes are both normal. So the father, therefore, got to be capital A, little a. So he mates with a genetically unrelated female, little a. And so that's where we end up with these different combinations. And in the Punnett square here, let me ask you this. So now, looking at the Punnett square, What is the recurrence risk? You got to know what the definition is, right? What is the recurrence risk that the probability will show pathology? Let me see your answers here. There we go. That's right. And the answer is it is a 50% probability of having the disease. 50%. This person and that person will have the disease because it's dominant, and these people here will not have the disease. So 1 in 2 possibility that the offspring will have the disease, okay, with autosomal dominant. Okay. All right. Now now that's usually the case. I want to stress, please be ready, because later on tonight we're going to be talking about something else. So with most autosomal dominant diseases, if they have 1 mutant gene, they will show pathology. One more time. With autosomal dominant diseases, if they have 1 mutant gene, of the 2 is mutated, they will show pathology. But as we will see a little bit later tonight, there are exceptions to this. And where somebody has a mutant gene and they have, with the autosomal dominant disorder, and they don't have pathology. And I'm telling you right now, the National Board loves those exceptions. Coming up later, don't worry about that now, but I'm just giving you a heads up. That's coming up a little bit later. Okay, now the diseases that are known to be autosomal dominant. By the way, if you want to know what's that about, it has to do with something called, not anticipation, Natalie, good try. It's called incomplete penetrance. Remember that incomplete penetrance? Remember the retinoblastoma? Oh, major stuff, major stuff. Did I say major yet? It's major stuff for board exams. Okay, but that's incomplete penetrance, coming up later with autosomal dominant disorders. But most autosomal dominant disorders, if they have 1 mutant gene, they will show pathology. But there are exceptions to this. Now, of the diseases that are known to be autosomal dominant, and there are many, these are the ones you should know without them telling you. You should know without them telling you. And these are familial hypercholesterolemia, that's the type 2A hyperlipidemia, the LDL receptor defect. You should know it's autosomal dominant. 1 mutant gene, and the patient will have the disease. Huntington's disease, neurofibromatosis type 1, we'll talk more about that disease later. Marfan syndrome, let me ask you this, in Marfan's, what's the name of the protein that's defective giving rise to Marfan syndrome? That's right, it's fibrillin, that's right. And you're going to see fibrillin for another reason coming up later on in the chapter to discuss another term called pleiotrophy. So you're going to see Marfan syndrome as an example, as an illustrator of autosomal dominant diseases on the boards, or you might see Marfan syndrome to illustrate the principle of pleiotrophy. Now, what's pleiotrophy? Stay tuned. There's only so much you can do at one time, but pleiotrophy is coming up later on in this chapter 1. Now, the other one here is acute intermittent porphyria. That is a disease that's associated with the porphyrias. Let me stress, you should know without them telling you that most porphyrias, most, not all, the one exception that I know of, but most porphyrias are autosomal dominant. So you should know that without them telling you. But there are many other diseases that are autosomal dominant that they don't expect you to know. For example, genes that cause hypertrophic cardiomyopathy. Those are genes that are involved in the sarcomere, remember the myosin heavy chain and the myosin binding protein. If you get mutations in those proteins, it's autosomal dominant, and you end up getting the hypertrophic cardiomyopathy, which is an autosomal dominant disorder here. It's a genetic disorder. In contrast to dilated cardiomyopathy, which is generally a vitamin problem like thiamine or alcohol or something like that. But the hypertrophic cardiomyopathy is autosomal dominant. But they don't expect you to know that. But I'm just pointing out there are many diseases they don't expect you to know, but these ones minimally know for boards. Now the next one to talk about here the modes of inheritance is autosomal recessive. So now what I look for with autosomal recessive diseases, again on the upper left-hand side, I start at the bottom and work your way to the top and ask yourself, Does it have a horizontal appearance? In other words, the disease in one generation, multiple people have the disease. But in other generations, nobody has the disease. So in other words, in other generations, it skips. The disease skips generations. And if you see that, horizontal appearance and disease skips generations, simply put, that's got to be autosomal recessive disorder. In contrast to autosomal dominant. Okay, now in these Punnett squares with autosomal recessive diseases, what do the letters stand for? And it's just the opposite of autosomal dominant. The capital A is the normal allele, little a is the disease allele. Please get those letters straightened out. You saw those letters earlier when we talked about cystic fibrosis. Remember all that in that pedigree and that clinical vignette. You saw letters there, you're seeing letters now, but you must master what the letters are referring to. If you see a pedigree or something like that, or you see a Punnett square on boards, you got to know what the letters are referring to. So just the opposite of autosomal dominant. Now with autosomal recessive, let's look at a particular mating of two people who are heterozygous carriers. And suppose the father is capital A, little a. Capital A is a normal allele, little a is the mutant gene. And in the mother or the female, she has a capital A and a little a. So both parents are heterozygous. Under that type of mating, what is the recurrence? Let me see your answers here. What is the recurrence risk of a mating of two people who have carriers for an autosomal recessive disease? That's right. It was 25%. Recurrence risk is 25%. That's right. And now let me ask you this, because I asked this yesterday, and a lot of you got it wrong yesterday. But what is the probability that the offspring will be a carrier? Let me see the answer here. What's the probability that the offspring will be a carrier? That's right. That's the 50%. That's right. Oop, back off here. Press the wrong button. 50%. So notice over here in this Punnett square, there's a 25% probability that the offspring will be capital A, homozygous normal. A 50%, 1 out of 2 possibility that the offspring will be a carrier, heterozygous for the trait, and a 25% probability of being homozygous with the mutant gene. And to express the disease, both genes must be mutated. Now, it turns out most genetic-based diseases, single gene diseases, most of them are autosomal recessive by far, or it's just due to missense mutation, something like that in a gene. So most autosomal recessive diseases deal with enzymes. So notice that now the 4 of them you see here are 4 you should know without them telling you, but again, there are many, many other ones. Sickle cell anemia with beta globin, cystic fibrosis with the CFTR, but many other ones are enzyme-based diseases. PKU is where phenylalanine hydroxylase is defective. Tay-Sachs disease, when the hexosaminidase A is defective, giving rise to those inclusion bodies and so on. But again, many other diseases are autosomal recessive. And the other ones, I doubt they expect you to know. They may say it on a board exam. They might say a particular disease like hyperprolinemia, comma, an autosomal recessive disease, comma, and then say something else about it. Always, always, on a vignette on board exams, if they give you the mode of inheritance, underline it, highlight it, make a mental note of what is the mode of inheritance. Because later on, we'll do a couple practice questions, and a lot of you will miss it. Will get it wrong because you're not noting the mode of inheritance, which they're telling you it is. I'll point that out to you later on once we do the vignettes. But the idea is that you should know that these 4 are autosomal recessive, and the other ones, I doubt they expect you to know. Okay, now what we're going to do is I'm looking at the time, and it's 6 minutes to the hour. So let's take a 10-minute break, and then I'll come back, and then we'll get into X-linked recessive, and a lot more to say about that one. Okay, take a 10-minute break, and see you shortly. Okay, everybody, welcome back for hour 3. And so what we've done so far with the pedigrees, the all-important pedigrees, you got to know them, is we talked about autosomal dominant, autosomal recessive, and what you need to look for on those particular pedigrees. So next, the third of the pedigrees to review is X-linked recessive. Now, on the upper left-hand part, what I'm going to do is list 5 characteristics of X-linked recessive diseases, inheritance here. And the first is, what does the word X-linked mean? It means that the gene of interest is located on the X chromosome. So if it's X-linked this or that, that means, don't get confused, don't make it complicated, that the gene of interest is located on the X chromosome. Okay, and another item is, because it's recessive, it has a horizontal appearance, and the disease skips generations. You start at the bottom, work your way to the top. Notice how in one generation there might be multiple people have the disease, but then there's another generation, nobody has the disease here. So and that's characteristic of recessive, in this case here, X-linked recessive. Another item here is characteristic is it affects mostly males. In other words, you see a pedigree like this on board exams, and everybody who has the solid, either square or circle, everybody who has the disease is the solid square. And so they're probably asking me about X-linked recessive because it affects mostly males. Notice the words, mostly affects males, because there are exceptions to where a female can have the disease, X-linked recessive disease. But it's rare, but it does happen here. I doubt the boards would go to that level of a low blow and give you an X-linked recessive mode of inheritance, and a female does have the disease, just to try to complicate things. I doubt they would do that to you. I know they can be pretty, the board people can be really ruthless sometimes, but that would be a little bit too ruthless to give you that kind of stuff. But it's possible. I mean, it's possible they could do that. All right, now another item here is that there is no male-to-male transmission in an X-linked recessive mode of inheritance. Now, it should be obvious to you, and I hate to ask the question, but I want to make sure. I just want to make sure, and that is why. Why is there no male-to-male inheritance with an X-linked recessive disease? Let me see your answer here, just to make sure, just so we don't get the it has to get the X from the mother. No. Why is there no male-to-male transmission? That's right, Tukir. That's right. There we go. That's right, Angela. There we go. Now we're talking. Now we're talking Turkey. There it is. Shireen. So that's right. And the reason is because the male, the father, is given to the son in male-to-male transmission. The father is given the son the Y chromosome. And if the gene is located on the X chromosome, then it's not coming from the father. Again, one more time. In male-to-male transmission of anything, remember, my Y chromosome came from my father. If you're a male in the audience, your Y chromosome came from your mother, from your father. So all the Y chromosomes that the males have came from their father. And and whereas the X chromosome that I have came from my mother, my Y chromosome came from my father here. So that's why there is no male-to-male transmission. Because, one more time, if the defective gene is on the X chromosome, because it's X-linked, then it's not coming from the father. It has to be coming from the mother, because the mother is given to the son the X chromosome, which may happen to have the defective gene. Now, again, this might sound obvious to many of you, but to some of you, you may not have thought about that. But while it's fresh in your mind tonight, go back over this and think, oh, that's right, that makes sense. You know, it makes sense. Just think about it. Don't wait longer, because it might get bogged down. While it's fresh in your mind, go back over it again until oh yeah, that's right, I should have known that, type of thing. So again, there's no male-to-male transmission. Okay, now what's coming up next is okay. Now what's coming up next is You see a pedigree. Now, again, no male-to-male transmission. As in this pedigree, let me show you. Here is a male who has a disease. Neither of his sons have the disease. Here's another male who has the disease. Neither of his sons have the disease. Here. And why? Because this father is given to this son. This father is giving to that son the Y chromosome. That's why they're males. And this father is given to his daughter the X chromosome. And so it's possible that he could be given to his daughter a defective X chromosome. But there's no male-to-male transmission in X-linked recessive disorders because it's a little bit of biochemistry information about the Y chromosomes coming from the father, not the mother. All right, now in a pedigree like this, suppose now notice here, they're not showing you who are because it's recessive, who are the people who are carriers. Here it comes now. I told you at the beginning of the chapter, we're going to practice this. And that is that to identify in a pedigree who are the individuals that are obligate carriers of a trait. In other words, they do one of the two genes, one of the two chromosomes has a defective gene, yet they have no pathology, but they are carriers of a trait, obligate carriers of a trait. No pathology, yet they have a defective gene. All right, now in fact, in this pedigree, there are 5 individuals. 5 individuals, in case they ask you what number of individuals are obligate carriers, or they might ask you who are they, that type of thing. So let's go through 3 of the 5 carriers are fairly easy to figure out. All right, let me ask you this. Which of these individuals in this complicated pedigree, which one got to be a carrier of a trait? That's right. And the answer is, it's got to be this great-grandmother, right? Now, I'm going to highlight them in the orange. In this case here, an orange circle. So again, the orange circle represents an obligate carrier. So now how do you know that's an obligate carrier, this female? Because her sons have the disease. Her sons have the disease, and the disease is not is not being inherited from the father because there's no male-to-male transmission, and it has to be coming from the mother. So the mother must be an obligate carrier. Who is another obligate carrier in this particular pedigree? Another easy one to figure out. And the answer is, it's this person over here. This female got to be an obligate carrier because the 2 sons have the disease. The son doesn't, because the son's inheriting the normal X chromosome from the mother. But the mutant X chromosome from the mother is being inherited by these 2 sons. So the idea is they got the disease from the mother. Got to be an obligate carrier. So the mother has 2 X chromosomes. One of them is normal, and one of them is mutated for a particular gene. The mutant chromosome is being inherited by these 2 sons from the mother. The good chromosome, X chromosome from the mother, is being inherited by the other son who does not have the disease. All right, now, anybody else in this pedigree? Anybody else? Here we go. This person right there, that female right there, got to be an obligate carrier. Why? Because one of the sons has the disease. So the son who has the disease is inheriting a defective X chromosome of the 2 from his mother and inheriting the Y chromosome from his father. That's why he's a male. This other son, his brother, is inheriting the good X chromosome from his mother and the Y chromosome from his father. So that's why this mother is an obligate carrier. Now, the other 2 obligate carriers are a little bit harder to figure out. We'll come back to those 2 obligate carriers in just a minute or 2. Now, with regard to X-linked recessive modes of inheritance, what do the letters stand for? And the letters stand for capital X versus little x. What the capital X stands for, it's the normal gene, the normal allele, the capital X. The little x stands for the mutant gene, the disease gene, the little x. So capital X normal, little x mutant. Please keep that in mind because you're going to, in X-linked dominant, it's going to be just the reverse. But X-linked recessive, capital X is normal, little x is mutated. Please keep those letters straightened out. Now let's look at a particular mating. And let me highlight the mating right here. Let's look at this male here. He's an affected male mating with a genetically unrelated female. So the male, his genotype of the sex chromosomes got to be little XY. The little x is the normal, excuse me, the little x is the mutant gene, the mutant X chromosome that he has. And his other sex chromosome is the Y chromosome, because he's a male. So his genotype of the sex chromosomes is little XY. So he mates with a genetically unrelated female, and because she's genetically unrelated, therefore she's going to be capital X, capital X. So she had no pathology, she's genetically unrelated, capital X, capital X. Under that type of mating, what is the recurrence risk? Let me see your answers here. Under that type of mating, what is the recurrence risk? And Drew, when you say how you differentiate between X-linked dominant, X-linked recessive, we haven't talked about X-linked dominant yet. I mean, you're jumping the gun. Wait till we get to X-linked dominant. We haven't talked about that yet. I know you want to know, and you're trying. That's great, and you're trying, but we'll get to that in just a few minutes. There's only so much I can cover at one time. Only so much I can do. So be patient with me. I'll get to that. You know I'm going to mention that, how you distinguish X-linked dominant with X-linked recessive. It's coming up after we talked about X-linked dominant, which we haven't got to yet. Okay, now, so in this type, what's the recurrence risk? And the answer is 0. There is no probability that any of the offspring will have the disease here. Okay, because now what's crucial here though, what's critical to know, here it comes, get ready. What's critical to know, under this type of mating where there's an affected male and a genetically unrelated female, what about the daughters? Look at the daughters. Notice that the daughters, let me highlight it in purple, all of the daughters are carriers. So this is how the National Board can ask this. If they say, who are the obligate carriers in this pedigree? It's these 3 right here, highlighted in the orange. But now the 4th one, got to be this daughter right here. The female offspring of that mating must be a carrier, because the Punnett square says so. Got to be a carrier. So now let's highlight it in the orange circle. So now we have 4. Who is the 5th one? Now take a quick scan of this pedigree. Who is the 5th one? Who is the 5th one? Who is the 5th one? Yeah. VR, not the sons. By the way, the sons are normal. The sons are normal. They inherited the good X chromosome from the mother and the Y chromosome from the father. So those sons are normal. They're not carriers. But the daughters are carriers. And who is another one that's an obligate carrier? And it's this female here, the sister of the other female who has the defective, who passed on the defective gene to her son. So this female there, because it's the same mating, affected male mating with a genetically unrelated female, this female there therefore must be an obligate carrier. So notice, there it is, the 5 individuals who are obligate carriers of a trait. So notice how 3 of them were easy to figure out. 2 of them, you had to know some little bit more information here. Again, you don't have to memorize that fact. All you got to do is just do a quick Punnett square. Just do a Punnett square of a affected male and a genetically unrelated female. You come up with this Punnett square. Those females are all capital X, little x carriers because they have the little x. Not hard. Now let's look at another mating here. Now another mating is, let's look at this one, where we have a normal male. Here's the normal male mating with a carrier female. She's an obligate carrier, so one of her X chromosomes is mutated. When they mate, then in the Punnett square, notice the Punnett square says the recurrence risk is 25%. 25%. But now on the females, 1 in 2 possibility of being a carrier, but they're not obligate carriers. So in other words, these 2 females right here may be carriers, but may not be carriers of a defective gene. Let me ask you this. How would you determine, if you wanted to determine exactly, precisely, these 2 females, if you were doing genetic counseling, of these two females, knowing it's possible they might be a carrier. Each of these two daughters might be this little quadrant here, or each of those two daughters could be capital X, capital X. So, but if you wanted to confirm it and be 100% certain, what do you have to do? What do you have to do? Let me see your answers here. What would you have to? That's right. That's the stuff we talked about in Chapter 7 in molecular biology. You do genetic testing, southern blots, and so on. Do the RFLP analysis to analyze the gene, the fragments from the gene by electrophoresis. Again, that's integration of genetics with molecular biology that we just talked about here. Okay, now coming up here is now other diseases that are known to be X-linked recessive. Again, there are quite a few of those. And the reason is the X chromosome is a huge chromosome. Lots of genes on the X chromosome, whereas the Y chromosome is very tiny. But in any case, we'll talk about that in karyotyping in Chapter 3. But the important feature here is of the diseases that are listed here, in my opinion, don't go memorizing these, but there are several of them that you should know without looking in the book or without them telling you. And that's, see this line here, this red line, is just put a line there. Everything of the diseases above it, you should know. The diseases below it, I doubt they're going to expect you to know that Menkes disease is X-linked recessive. It is, but I doubt they expect you to know that. But you certainly should know that Duchenne's G6PD deficiency. You should know that hemophilia A affects males. By the way, hemophilia A, what protein is defective? Hemophilia A, what protein is defective? That's right. If you don't know the answer, you always say factor VIII. Hemophilia B, which protein is defective? Hemophilia B. That's right. That's factor IX. That's right. So in hemophilias, they affect mostly males here. Okay, now what's going to come up next is, before we get to X-linked dominant, what I want to do is explain something here, because a lot of times students have a hard time understanding this. Is suppose I inherit a mutant gene on the X chromosome that I got from my mother. Okay. And as a result, I'm going to have the disease. Yet my sister, if I have a sister, and I do, but if my sister inherits the same X chromosome with the mutant gene, she does not have the disease. Okay, that's what we're going to try to figure out. Simple. I'm making it simple, Natalie. Making it simple. So here comes the issue again. What we're trying to review here is that if my mother is a carrier female and she's given me one of her two X chromosomes, and it happens to be the X chromosome that has the mutant gene, I'm going to have the disease. I'm going to have pathology. But my sister, who also inherits the same mutant gene, she does not have the pathology. Yet she has the defective gene. So that's what we're going to try to analyze from a biochemical point of view. So the idea is, and that's shown here on this issue of X chromosome inactivation, a little review of X chromosome inactivation. Now what this is about is if a female inherits twofold the number of genes as males, because there's two X chromosomes in a female. A male has one. How do the females prevent twofold number of enzymes or proteins? Because even though they have two X chromosomes, the genes on them are not being expressed at the same time. Only one of the X chromosomes is being expressed. And everybody knows the reason for this is that one of the two X chromosomes in the female is condensed into heterochromatin, and the genes are turned off by cytosine methylation to form the bar body. So the bar body is where one of the X chromosomes of the two in a female is turned off by cytosine methylation. The genes are turned off, and the chromosome is then condensed all the way to pure heterochromatin, and genes are never turned on for the rest of their lives. Here. By the way, what's this process called? This process of X chromosome inactivation. What is the other name for it? You might see another name for X chromosome inactivation. What's it called? That's right, for me. That's right. Well done. X-ray. Not imprinting. No. That's imprinting something else at the end of the chapter. Don't worry about imprinting now. This has nothing to do with imprinting. And the answer is, that's right, it is lionization. So lionization is the same thing as X chromosome inactivation. It's the process of taking one of the X chromosomes in a female cell and shutting down that X chromosome by cytosine methylation, forming entirely heterochromatin. All right, now the idea here is when does this lionization take place in a female embryo? Suppose we have now a female embryo, and this is a blastocyst. Now remember from embryology that you had with Dr. White, is that in the blastocyst there are about 100 cells at day 16. And at day 16, that is where the lionization starts to take place in a developing embryo. And in that female embryo, remember these cells are embryonic stem cells. They have not yet differentiated into somatic cells and so on. They're not differentiated yet. But at about day 16 is where lionization takes place. And what happens is, suppose in each of the cells there are two X chromosomes. One of the X chromosomes came from the father, and the other X chromosome came from the mother. But in 50% of the cells, in half the cells, in this example, there are 50 of them. In 50 of the cells, what happens is one of the X chromosomes that came from the father is turned off by cytosine methylation, forming the bar body, or the other X chromosome is active, coming from the mother. In the other half of the cells in this embryo, the chromosome that was inherited from the father is active. The other X chromosome that was inherited from the mother is inactive, or the chromosome is lionized to the bar body. So this is now, one more time, let me spell it out. In one half of the cells of a female embryo, one of the two X chromosomes is inactivated, and the other cells, the other X chromosome is inactivated. This process, lionization, is random and it's fixed. Never to be, you don't take a bar body and go back to the active chromosome. It's random and it's fixed. And the idea is it should be 50-50. So in other words, it's in 50% of the cells, the X chromosome inherited from the mother is active. In the other 50% of the cells of the body, the X chromosome inherited from the father is active. Now remember that these cells, these embryonic stem cells, will then differentiate into skin cells, brain cells, liver cells, and in the body they'll differentiate into 10 trillion cells. In the body right now, you have 10 trillion cells that came, if you're a female, that came from this 100-cell blastocyst here. So, and then half of the cells, one X chromosome is active, and the other half of the cells, if you're a female, the other X chromosome is inactivated. 50-50. Now, you might see the term called X chromosomes skewing, or something about skewing of the X chromosome. And what that has to do with, in some situations, it's rare, but it's possible that instead of being 50-50, random issue, it might be 51-49%. So in other words, in some cells, in the cells in the body, they may have a little bit more of the X chromosome being inherited from the mother's on, and a little less of the X chromosome coming from the father. That's called skewed X chromosome inactivation. But I wouldn't worry too much about that. That's a much lower item here. Okay, now coming back to the issue, then of pathology. So if a defective gene is located on the X chromosome, then why doesn't the female, if she has a defective gene on one of her X chromosomes, why doesn't she have pathology? Now what we're going to do is we're going to talk about this in real life terms regarding a disease called Lesch-9 syndrome. Now you know Lesch-9 is X-linked disease, X-linked recessive disease. Let me ask you this without looking in the book, without looking at the previous pages and so on. Don't look. I'm trying to see if you can recall. What's the name of the enzyme when it's defective gives rise to Lesch-9 syndrome? What's the name of the disease? The enzyme that's defective. That's right. Major enzyme for board exams. Major enzyme. But don't worry about the details now. It's all coming up later on in metabolism. In fact, late next week. But and let me ask you this. What metabolic pathway is HGPRT in? What pathway is it in? That's right. It's involved. It's in purine metabolism, but specifically purine salvage. It's the main enzyme that salvages purines. So the idea and so in Lesch-Nyhan syndrome, a female has defective gene. She doesn't have pathology, but a male does. So how do we explain that? To explain this, suppose I'm going to color code this. Suppose in the blue rectangle that represents the mutant gene for HDPRT, and the red rectangle represents the normal gene for HDPRT. Now suppose in this example, the mutant gene is being inherited from the X chromosome coming from the mother. So the mother is given to the daughter the X chromosome that has the mutant gene. So in the embryo, in the blastocyst here, in half of the cells in the body, the gene that's mutated on the X chromosome inherited from the mother, there it is. Whereas the gene that's normal, being inherited from the father, it's cytosine methylated, inactive in the bar body, never to be turned on. So these cells, and half of the cells in the embryo, they're making no functional enzyme. They cannot salvage purines here because they have no functional enzyme. Whereas in the other 50% of the embryo cells, the X chromosome coming from the father is active. And that's the chromosome that has the normal gene for HDPRT. So it's making normal enzyme to salvage purines. Whereas the mutant gene is shut down in lionization because it's inherited from the X chromosome coming from the mother. So what this means is, so again, one more time. So in 50% of the cells, they're making no functional enzyme. In 50% of the cells, they're making good enzyme. Let me spell it out again. Here it is. In one half of the cells, they're making good HDPRT. Right? Because right there. So half of the cells of the embryo, and therefore half the cells in the body when the embryo differentiates into the 10 trillion cells in the human body. In half of the cells, in 5 trillion cells, they have good enzyme because they have good gene. The mutant gene is turned off. But in the other half of the cells in the body, 5 trillion cells, they're making mutant enzyme, which is right there. They're making the mutant enzyme, and the other X chromosome that has a good gene is turned off. But the important feature here is in the entire body, there's enough good enzyme being made that they can salvage what purines are normally being made. Remember, this is an enzyme, and the purines, when they start accumulating, can get into the blood, and they could get into other cells, and if they have the good enzyme around, they get into those good cells, and they can be salvaged. So in the human body, they have good enzyme, enough good enzyme to salvage the purines, causing no pathology. Whereas in a male, if he inherits an X chromosome that has defective HDPRT, 0 cells in the body, none, have good gene. Therefore, none of the cells have good enzymes. Therefore, they can't salvage purines. Therefore, they end up with all that gout and with the severe biting of the fingers and toes, and we'll talk about it later on. But there it is. This is the biochemical explanation behind the genetics here. You can memorize all you want, but unless you understand why, there it is. It just makes sense. Okay, now what's coming up next is the fourth of the modes of inheritance, and that is X-linked dominant. Okay, forget who was the one asking about Anjou, I think. But in any case, now on the upper left-hand part, here is the issue of 5 characteristics of X-linked dominant mode of inheritance. One characteristic is it's the gene of interest, again, is located on the X chromosome because it's called X-linked. A second characteristic is because it's autosomal dominant, it's X-linked dominant, it's a dominant disease. It has a vertical appearance, and the disease is in every generation. Just like autosomal dominant, X-linked dominant has a vertical appearance. The disease is in every generation. Another characteristic is that there's no male-to-male transmission for the same reasons we mentioned with X-linked recessive, because the father is given to the son the Y chromosome, not the X chromosome. And then the other characteristic of this one is with X-linked dominant disorders is that the affected male transmits to all of his daughters. Now again, if that was Anjou, I can't remember who it was, saying how do you distinguish X-linked recessive from X-linked dominant? These are right there, right there. The disease is in every generation, doesn't skip it, and affected male transmits to all of his daughters. And another characteristic, if you want to write this down, notice in this pedigree here, a quick scan of it, you see a lot of females have an X-linked disease. There it is. Whereas with X-linked recessive, it's mostly males. Okay, so there it is. So again, it's not hard to figure this one out either. Okay, now regarding this term, affected male transmits to all of his daughters. In a Punnett square, let's look at a particular, now what do the letters stand for? Again, with X-linked dominant, capital X is the disease allele, the mutant allele, little x is the normal allele, just the reverse of X-linked recessive here. So you got to get these letters straightened out. Now let's look at a particular mating here. Suppose we're looking at an affected male mating with a unrelated, genetically unrelated female who doesn't have any kind of disease here. Under that type of mating, again, the male is capital X because it's disease allele, not little x, it's capital X, the disease allele because it's dominant. Capital XY, that's the male, and the female is genetically unrelated, little x, little x. So therefore, under this type of mating, notice the recurrence risk. Recurrence risk is 50%. But importantly, notice under that type of mating, notice here, right here, see that? All of the daughters, there it is, all of the daughters right there, as it states right here, all of the daughters has a disease. Okay, all the daughters, so in other words, there it is, all the daughters have the disease here. So if there was a fetus and it was it was found that the fetus is going to be a female, under that type of mating, that female will have the disease because all the daughters have the disease. All right, now so let's look at another mating. And that mating is where we have a normal male, genetically unrelated male, mating with a heterozygous affected female. So one of her X chromosomes in the female, in the mother here, has a defective gene. Her other gene, other X chromosome is normal. Yet when they mate, and again, so it's going to be little x normal, Y in the male, normal male, mating with a heterozygous affected male, capital X, disease, little x normal. Under that type of mating, the recurrence risk is still 50%. Just like that autosomal dominant, 50% risk. Same thing here with X-linked dominant, 50% risk in the Punnett square. But notice as well, there are daughters that do not have the disease. So that's why it has to be an affected male transmits to all of the daughters, not an affected female. An affected male transmits to his daughters. All right, now another item here is the diseases that are known to be X-linked dominant. It turns out there are not that many. Only about a half a dozen diseases are known to be X-linked dominant. And most of those few that are out there, forget about it. I got to ask. There are only two that are important to know of the half a dozen that are known to be X-linked dominant. And of those two, one is called the hypophosphatidec rickets. This is sometimes called the vitamin D resistant rickets. Remember, rickets is often caused by a nutritional deficiency of vitamin D. But these people have rickets, you give them vitamin D and it doesn't work. So that's a genetic, not a nutritional problem. And so that gives rise to hypophosphatidec rickets. But much more important than that disease for boards is Fragile X. You must know, without them telling you, it is an X-linked dominant disorder. And so board exams, if I ask what's an example, which of the following diseases is an example of X-linked dominant disorders, and you look for Fragile X. By far the most important disease. Let me stress as well, I keep emphasizing over the last several days, some of these diseases you got to know for multiple reasons, not just the pathology. Yeah, you got to know the pathology, but some of these diseases you got to know for other reasons besides the pathology. For example, We talked about Tay-Sachs disease. You got to know its pathology with a cherry red spot in the macula and psychomotor retardation, the pathology. But you should know it as well because it's an example of an insertion disease, an insertion of 4 nucleotides causing the frame to shift. So it's an insertion disease. Also, because the frame shifts, it messes up the splicing. So it's also an example of a splicing disease here. And as we'll talk about in the next chapter, you're going to see Tay-Sachs disease for yet another reason. And that's the issue of the Hardy-Weinberg, the use of it in the Hardy-Weinberg equilibrium. So you got to know some diseases for multiple reasons. But here is Fragile X, another example. It's why it's so high yield, just like Fragile X, just like Tay-Sachs, just like osteogenesis imperfecta. You got to know it for multiple reasons. As we talked about the other day, you're going to hear more about OI in this chapter. It illustrates a principle of something called locus heterogeneity. What does that mean? Stay tuned, coming up later on in this chapter. But the idea is Fragile X. You got to know it not only for the pathology. You know what the pathology is, the second most common cause of mental retardation in human beings. It's ranked below what? What's the most common cause of mental retardation? That's right, it's Down syndrome. Down syndrome by far, not by far, but it's Down syndrome is the most common cause of mental retardation, and actually not that far behind Downs in terms of mental retardation is Fragile X. Now, in addition, facially, they have those protruding ears and have a characteristic elongated face, something along that line on board exams, elongated face, protruding ears, mental retardation. I say to myself, well, they're probably asking me about Fragile X. But yet another reason why you need to know Fragile X. What's another reason? Let me see your answer here. What's that? We talked about it earlier, so let me see if you can remember this. What's another reason why you might see Fragile X on the boards besides the pathology and besides its X-linked dominant? What's another reason? We talked about it earlier. I'm trying to see if you can recall it. We talked about it. Not an insertion of what? That's right. It's a 3 nucleotide insertion that does not cause the frame to shift. It's one of those 5 examples of a triplet expansion repeat disease. Well done, well done. Those of you who write that down, well done, excellent job. You're remembering. So again, always try to think how they're going to ask you. They might ask you about Fragile X for a number of reasons, not only with the pathology. It might be a double jump question, triple jump, where in the vignette, you have to figure out it is Fragile X, but they're not asking you what's the name of the disease. They're going to ask you based on your ability to figure it out. Well, then what's the cause of the mutation or something along that line? How can it be analyzed? So those type of things. So again, major issues of how they can ask questions on boards. Okay, now another item here. So we've done 4 of the 5 pedigrees. Now the last pedigree to talk about is equally important for the boards. Even though there's not that many diseases, I'm telling you right now, the National Board loves mitochondrial inheritance. Even though it's diseases are rare, they love it for boards. Don't equilibrate, don't equate common diseases or something like that, frequent diseases with the possibility of being on boards. They love mitochondrial inheritance. Every year, and sometimes multiple times, I hear about mitochondrial inheritance diseases on boards. So please be ready to understand mitochondrial inheritance diseases. Matter of fact, I hear about it every year, and about a year ago or so, a student told me they got mitochondrial inheritance. And what he told me he saw on the board exams, he said he got it wrong, unfortunately. But what he told me he saw on the board exams, what they were asking him was astounding. I could not believe it. When the student first told me, they asked about this. I said, man, why were they asking that? That's such a minor item, trivial item. Why were they asking it? And then it dawned on me. Oh, now I know. Okay, now I know. And so I'll tell you what I heard. Just stay tuned. Be patient with me. I'll tell you what that student told me, that you need to be prepared here. Now, first of all, mitochondrial inheritance. The characteristics of mitochondrial inheritance it has a vertical appearance, and the disease is in every generation. So here it is. You start at the bottom, work your way to the top. Notice how in one generation, multiple people can have the disease, and it's in every generation. Another characteristic is there is no male-to-male transmission. Here's a male who has a disease. None of his sons have the disease. Let me ask you this. Thus far, and you, I need you now because I want you to distinguish this. Thus far, what does this mode of inheritance sound like? Which of the other four modes of inheritance do these two comments up here sound like? What does it sound like so far? That's right. It sounds like X-linked dominant, right? But the main difference now, here it is. This is the distinguishing feature, that the disease is inherited only from the mother. And the reason for that is because mitochondria have DNA, and mitochondria therefore have genes. And these genes can be mutated, causing pathology. So if a mother has a defective gene in one of her mitochondrial DNAs, and she's going to pass on that defective mitochondria onto her offspring, every single offspring will have the disease. So mitochondrial inheritance actually is very easy to analyze. All you got to do is when you see a vignette or a pedigree like this, you look at it and ask, if the mother is affected, then all of the children, male or female, all of the children have the disease. Here's a female has a disease, all the offspring have it. Here's a female has a disease, all of the offspring have it. But now, if the male has the disease, here's a male who has the disease, no children are affected, because the mother is given to the offspring, male and female, mitochondria. And mitochondria have the DNA, and if it's defective, it's coming from the mother only. The father is not given mitochondria to the offspring here. So it's very easy pedigree to figure out here. Not that difficult here. All right, now, how might they ask this? What kind of items do I hear about mitochondrial inheritance? Well, what they could do on board exams is they can give you a pedigree like this. And they say, they give you all the information about the pedigree, and they might ask you, might integrate this with anatomy or histology. They might say, based on a pedigree like this, an offspring who are a family member from this family, which of the following tissues might be adversely affected? One more time. A person who comes from a family such as you see in front of you here, this pedigree, which of the following tissues might be adversely affected? In other words, these tissues won't work very well. And you would say to yourself, well, because these are mitochondria issues, that means any tissue that's highly aerobic, like neurons, like muscle cells, will be adversely affected. Matter of fact, the three known diseases that are known to be mitochondrial inherited are Leber's neuropathy, which is a neurological problem, neuron problem, and MELAS and myotonic epilepsy with ragged red muscle fibers, sometimes called MERF. These are these mitochondrial issues as well, muscle issues as well. So highly aerobic tissues would be highly affected. Now, another way they could ask this on board exams is they might say to you, based on a pedigree like this, and a person coming from a pedigree like this, which of the following pathways, biochemical pathways in metabolism, might be adversely affected? And the way you answer this, because you're a thinker, you're not memorizing here, you say to yourself, well, if mitochondria is where you have a defective gene, then likely pathways in the mitochondria would be affected. And sure enough, is that electron transport chain might be affected, oxidative phosphorylation might be affected. Matter of fact, Leber's neuropathy, I doubt you have to know this, but the biochemical defect in Leber's neuropathy is oxidative phosphorylation. I doubt they expect you to know where the defect is, but it is what's causing Leber's neuropathy. is the electron transport chain oxidative phosphorylation is not working like it should. Okay, now, again, I hear about mitochondrial inheritance on boards quite frequently. But this question, this vignette that another student told me about a year ago or so, astounded me. And you got to be ready for it, because what it means is it's in their database of questions on the USMLEs. And I have no idea how many questions are in their database. Nobody knows. Whether there's 1,000 questions, whether there's 5,000 questions, 10,000 questions, I have no idea. Nobody knows how many questions, vignettes are in their database. But when you go take that national board exam, the computer that you're sitting in front of, the computer, no, the laptop is in front of you. But in any case, the computer is going to give you 322 questions from that database. And one of the questions deals with this topic coming up here. The student said that he got a pedigree that he quickly analyzed as mitochondrial inheritance. And they didn't ask what was the mode of inheritance, they asked which of the following molecules would be adversely affected. Not pathways, whatever. He had which of the following molecules would be adversely affected. So he thought, well, if it's mitochondria, going to be electron transport chain, oxidative phosphorylation. So he was looking for an option that had something like cytochrome C, complex 4, something like that. He was looking for one of the components of electron transport chain. But none of the options had to do with that. And he missed it because he didn't know. So here is the answer. And be ready with your highlighter, because it's in their database of questions. Because what the boards was asking is that they expect you to know that some of the tRNAs, 22 of them, and some of the ribosomal RNAs, 2 of them. Don't worry about the number here, but just all you need to know is some of the tRNAs. Remember, there's at least 61 different tRNAs because there's 61 codons. Some of them are encoded by mitochondrial DNA, and some of the ribosomal RNAs are not encoded by the nucleolus. They're encoded by mitochondrial DNA. You probably don't remember this, but the other day when I asked you in chapter 3, where are those 3 kinds of RNA being made? And I said they're mostly made here in the nucleolus or in the euchromatin. Remember all that? Probably don't remember, but I did emphasize they're mostly made here because I knew I would be talking about this. That some of the tRNAs and ribosomal RNAs are made from mitochondrial genes. Now, so when the student told me that, man, I thought, wow, why were they asking that? That seems like a low blow, you know? I mean, I know these people are not your friends, but man, why they ask a question like that? And then it dawned on me. Because the other 2 diseases that are known to be mitochondrial inherited, I doubt you have to know again, this is low yield part. I doubt you have to know this part, but the other 2 mitochondrial diseases, MELAS and MERF, one of them, that's MELAS, is due to a defect in the tRNA for lysine, and MERF is due to a defect in the tRNA for leucine. Don't worry about that. That is, I mean, I doubt they're going to ask that, but I'm driving home the point that the tRNA, what they're expecting to know is that some of the tRNAs are made by the mitochondria genes, and if they get defective is going to give rise to these diseases here called MELAS or MERF. That's what they expect you to know. So don't overcomplicate this. But the idea is make it simple, understanding what goes on. And again, you must know mitochondrial inheritance. Okay, so right now I'm looking at 8 minutes to the hour. So what we're going to do is we're going to take a 10-minute break, and then I'm going to come back, and then we're finished with all 5 modes of inheritance. And now we're going to do the remaining part of the chapter is all these exceptions and complications to the normal modes of inheritance. So let's take a 10-minute break, and then we'll move on. Okay, everybody, welcome back to the last hour in genetics here. And where we're at is in chapter 1, and we're talking about all 5 of those modes of inheritance, which you must be able to interpret, how to analyze them as either as questions on their own or by using the pedigree information to answer another question that they might ask. Now what happens, now what's come up for the rest of the chapter? There are a series of terms, all kind of maybe a half a dozen different terms coming your way. So with these terms, I'm going to give you the definition of what the term means, and then you should always know at least one disease and pathology that's associated with that term. One more time. So there's going to be a lot of terminologies coming up in this chapter, the rest of the chapter, and you got to know what it means. I'll give you that definition, and then what's an example of a disease that illustrates the principle of that term. So the first term we want to talk about is variable expression. Now before I give you the definition, what do you think it means? I mean, you can memorize, but what do you think variable expression means? What do you think that means? What do you think that means? I mean, it's not hard. Just use your mind. What do you think it means? That's right. Variable, what does it mean? It means that in a pedigree, everybody who has a defective gene, some people are severely affected and some people are mildly affected. Variable expression. One more time. Variable expression means some people in a pedigree who have a disease are severely affected, other people are mildly affected. Now, again, don't look in your book. The book has the answer. I'm just trying to get you maybe you didn't. Let me straighten it out here a minute. Maybe you didn't know this. Maybe you didn't. But let me be the first to tell you, if you didn't know this already, that when you take the National Board exam, you're not, let me underline the word not, permitted to have a book in front of you. You're not permitted to do a Google search when you take the National Board exam. Okay, so maybe you didn't know that, but I'm just telling you in case. Nobody ever told me that. I'm telling you, you're not allowed to have a book. Okay, so the idea is that's what I'm trying to do here. Is if I ask you an oral question like I just did, don't go looking on what's the answer. It's in the book here. Let me type it up, and it does nothing. It does nothing. It's not. You're not fooling anybody. All I'm trying to do is to try to see if you can think of something. Oh yeah, what does it mean? Just follow. Just try to follow the reasoning, and you'll get to the answer. So variable expression, what it means is this is one of the several complications and exceptions to the typical mode of inheritance. And variable expression means that every diseased person in a pedigree has the disease, but to different extents. In other words, if you see a pedigree where maybe 3 or 4 people have a solid circle or solid squares, but that one person might be severely affected and another person is mildly affected. Variable expression. But I want to emphasize that they do have some pathology. They have some phenotypic expression. Again, you must know this, so variable expression, some people are severely affected, some people are mildly affected, but they have at least some pathology. Why is that so important? The some here, some phenotypic expression, because the next term, the next definition coming up shortly is going to be a term called incomplete penetrance. And there they have no phenotypic expression here. But in variable expression, they have some pathology. All right, now I'm going to give you an example. Okay, so they ask you, in fact, many diseases show variable expression. Now, one great example is neurofibromatosis type 1. Again, Dr. Barone will get into the pathology of that disease and so on. It's a variable number of tumors and sizes of tumors. Now, let me show you an example of a person who is severely affected by the defective gene. And this person clearly is severely affected. Many solid tumors and clearly severely affected. But his brother may have just maybe 2 or 3 tumors, something like that. So it is variable. They both have tumors, but this person clearly is severely affected. Now, the name of the gene responsible for neurofibromatosis type 1 is an easy way to remember that. It's NF1. Neurofibromatosis 1 is the gene on chromosome 17. Now you're going to ask, and rightfully so, do you need to know the chromosome number? I doubt. I think that's low yield. So I'll show you some chromosome numbers coming up with some of these other diseases, but I wouldn't worry so much about the chromosome numbers. Now, some chromosome numbers are absolutely critical to know. Chromosome 15, that should be ringing bells in your mind right now. Chromosome 5, that should be ringing bells in your mind right now with a certain pathology. But here, NF1 is located in chromosome 17. I doubt they expect you to know that. Now, the issue here is, let me ask you this. What class of protein is the protein encoded by the NF1 gene? What class of protein is the product of the NF1 gene? Is it a kinase? What kind of class of protein is it? Receptor? What is it? I think we talked about this the other day. What is it? Receptor? No. We talked about it on the way back. There we go. Now we're talking. Now we're talking here. That's right. It is a tumor suppressor gene. A tumor suppressor gene. Remember the issue that it's like the P53, like RB and all these things. These are proteins that kill a cell by apoptosis. And if the gene for that protein is defective, then the cells will go into S phase. They'll start multiplying. In this case here, to making many solid tumors, okay, in neurofibromatosis type 1 here. Now, another item here is the issue of the mode of inheritance. In neurofibromatosis type 1, we talked about it at the earlier part of the chapter, and it is an autosomal dominant disorder. Here is neurofibromatosis type 1. Okay, so this is variable variable expression. Sorry. I apologize for being distracting like that, but can't help it. But okay, so anyway, now the next term to talk about. So again, variable expression, you know an example, you know what it means, there it is. Now the next example of a term you need to know is a term called incomplete penetrance, which is even more high yield. This is one of the most important terms in genetics that you must master, incomplete penetrance. Now, what incomplete penetrance is about is, this is where they have a disease-causing mutation, and when you think incomplete penetrance, you think autosomal dominant disorders. Technically, it could be for autosomal recessive disorders, but in my opinion, don't worry about that. Okay? Make life simple. Don't overcomplicate. So when you think incomplete penetrance, you think autosomal dominant disorders, and then you'll start to understand what's going on over here. So think autosomal dominant, and that's the way they're likely to ask you about incomplete penetrance on the boards. Now, the idea here is that, remember, with incomplete penetrance, that back off. When we talk about autosomal dominant, that means a person has inherited one bad gene and one good gene. One bad gene and one good gene. And because it's autosomal dominant, they should have pathology. But in this case here, with incomplete penetrance, that person has no pathology. The word no, highlight the word no. They have no pathology. Even though they have a defective gene, they have no pathology here. Okay, now in this pedigree here, what kind of mode of inheritance is it? So you look at it, okay, it has a vertical appearance. The disease is in every generation. Therefore, it is autosomal recessive, and there's male-to-male transmission. How do we know that? Because for these two males right here to have the disease, the grandfather had the disease. He's passing it on to his son, who then passes on the defective gene to his sons, giving rise to the disease. So the incomplete penetrance is clearly this person here. So that person there must be an obligate carrier of the trait. And he should have the disease with autosomal dominant, but he doesn't. Okay. All right, so everybody with me so far? I want you to know this because it is high yield. Okay, everybody with me? If you get, type in your questions to Dr. Seda and Dr. Donald. They are happy to answer your questions. They can't give you a thesis for an answer, but they'll have to be brief and get right to the heart of it. All right. Please repeat. Okay, with incomplete penetrance. Again, this son here, this father of these two children, these sons who had the disease, must have a defective gene. Why? Because the mother's not giving these two sons the disease. It's got to be the father. And this father is inheriting the defective gene from his mother. And with autosomal dominant disorders, we go from the grandfather to the father to the sons. And that would be the normal issue of autosomal dominant. But in incomplete penetrance, it says, what's the definition? It says they have a defective gene. One defective gene, one good gene. They should have pathology, and they don't. So this son does not have disease. And so that illustrates the principle of incomplete penetrance here. Okay, I can't keep repeating everything because I'll never get done. There's a lot of content coming your way. If I have to repeat everything 5 times, it ain't gonna get done. Next Friday, I'll be on what an amino acid is. So we have to keep repeating things. So please pay attention. If you have questions, ask on the Q&A pod. Okay. Now what's coming up next is a terminology that's related to incomplete penetrance. Now pay attention, because this one could be a little tricky if you're not paying attention. And this term is called degree of penetrance. Now, degree of penetrance means that it's a term, it's a percentage. It's the number in the numerator. It's a number of individuals who show a disease, who show pathology, who have a clinical manifestation because of a defective gene, divided by the number of people in a pedigree that have a defective gene. So one more time. Degree of penetrance means it's a number of people in the numerator who have pathology divided by the number of people who have the defective gene. Now, most autosomal dominant disorders are 100% penetrant, meaning everybody who has a defective gene, everybody has pathology. LDL receptor deficiency, for example. LDL receptor, you remember the type 2A hyperlipidemia, you have a defective LDL receptor gene, you're going to have pathology. Your cholesterol number is going to be 350 or something like that. So again, degree of penetrance, number of people who show pathology divided by the number of people who do not, who have the defective gene. So most autosomal dominant disorders are 100% penetrance. But the exception is here with incomplete penetrance. If on board exams they say to you, a particular disease is 90% penetrant. What does that mean? That means that there are, in a pedigree, there are 90% of the people show pathology, and they're divided by the number of people who have a defective gene. So 9 out of the 10 people in a pedigree have pathology, and they have the defective gene. And there's 10% of the people in a pedigree do not show pathology, even though they have a defective gene. All right, let me ask you this. See that pedigree in front of you there on page 314? If you were asked on board exam, what is a degree of penetrance of this particular disease? Whatever this autosomal dominant disease is, whatever it is, doesn't matter. But what is a degree penetrance here? Let me see your answer here. There you go, Anju. There you are. See, Anju, man, you're thinking, man. That's great. That's absolutely great. That's right. And the answer is, how many people have the disease? Let's count them out, as shown by the pedigree. 1, 2, 3, 4, 5, 6. So the numerator is going to have 6. How many people have a defective gene? Count them out. 1, 2, 3, 4, 5, 6. And this person here has a defective gene. Even though he has no pathology, he has a defective gene. How do you know that? Because the sons have the disease. If the sons have the defective gene, where'd it come from? Either has to come from the mother or the father, and the mother is genetically unrelated. So it has to be coming from the father. So even though he has no pathology, he has a defective gene. So the degree penetrance, not hard, 6 divided by 7, turned out to be 86%. There it is. That ain't hard. It ain't hard. Okay. If this is hard, I don't understand this. You're maybe either overcomplicating this or being distracted or whatever. It's honestly,
It's not that difficult if you just think. Look at the words. Okay. Now, why is this so important for board exams? Because remember, with autosomal dominant disorders, that you inherit one mutant gene and one normal gene. And with autosomal dominant disorders, the person should have pathology. But in incomplete penetrance, again, let me show it with these two hands. One good gene, one bad gene that a person inherits. And because it's autosomal dominant, they should have pathology, but they don't. But now, in a somatic cell, if there's a second hit that destroys the good gene in one cell, one somatic cell, then in that single cell, then that cell is now, both genes are faulty, both genes are defective, and it'll set off the disease. And so again, this is the second hit issue. So if there's a secondary mutation in the somatic cell, it'll set off the disease. Here, one more time. One good gene, one bad gene, you should have pathology, but you don't. But if there's a second hit on the good gene that just kills it, like what? Like what's going to be a second hit? Like you go to the airport and you get all that X-rays or whatever it is, that the metal detectors, or you go to the dentist and you get X-rays bombarding, your teeth and all that. Those are potent things that could damage DNA, and that could set off it. That X-ray could actually kill a good gene, and the other gene is faulty to begin with genetically, could set off the disease. Now, why is this so important for board exams? Is because there are a number of diseases, well-known diseases, that are known to be show incomplete penetrance. And by far the best example they hammer medical students on, as I mentioned earlier, is retinoblastoma. So again, this picture, based on the biochemistry I just mentioned to you, this picture in the retina is somebody has retinoblastoma. That is a normal person, I should say. Even though they have one good gene, one bad gene, they should have pathology, but they don't. But in the retina, if a photon of light, and photons or electrons, are bombarding the retina and so on, and a photon then hits the good gene and absorbs the light, and DNA might break or something like that, damage that gene, that particular gene, and in that single cell, both genes are defective. One's due to genetics, the other is to that environmental second hit, and as a result, that cell now, remember, what is RB supposed to do? Well, let me see your answer here. How do you connect the dots? Well, okay, so both genes will be defective. Well, why do they have a cancer? Why do they have that retinoblastoma? Why? Because what is RB supposed to do normally? What's the normal biochemistry of the RB gene? That's right, it's a tumor suppressor gene. That's right, we talked about those earlier. So the idea is, so now you kill a tumor suppressor gene in one cell, and then as a result, those cells, those retina cells, make 2 cells, eventually mitosis, 4 cells, and eventually you end up with a retinoblastoma. Again, not hard here, just understanding here. Now, other examples, Lynch syndrome, hereditary non-polyposis colorectal cancer, hemochromatosis. Matter of fact, hemochromatosis, its degree of penetrance is something like 35%. It's very low. In other words, a lot of people, it's an autosomal dominant disorder, iron uptake. And what happens there is people should have hemochromatosis, but a lot of people don't, because even though they have a defective gene, they're fine. But then if that second hit occurs later in life, it's going to set off the disease, hemochromatosis. And in addition, these other tumor suppressor genes, BRCA1, BRCA2. So again, you must know the definition. You must know what it means here. You must know how does it occur, and pathological examples of that particular term. In this case here, incomplete penetrance. Now the next term to talk about. Okay, everybody with me? All right, everybody, please say, okay, good. You're with me. Okay. Because a lot of times I see students, it's going too fast or going, everybody okay? Everybody good to go? Okay, good. All right. Because I don't want to bore people. I mean, if I keep repeating things, there's people that get it, and then I bore them to death, and they're yawning at me, and I'm a very sensitive guy. When people start yawning at me, and I'm doing the best I can, I'm sweating, I don't feel good right now, and I'm doing the best I can, and people are yawning because they're bored stiff. I'm very sensitive. So the idea is I got to go at a pace that doesn't bore some people and hits home with other students. So it's part of this issue. All right, now the next term we need to talk about is pleiotrophy. Now what pleiotrophy means is that if you see the term on board exams, you might see it as an option of a vignette. And they might say this disease, blah, 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 shows which of the following principles. It might say incomplete penetrance, option A, option B is pleiotrophy, something like that. What pleiotrophy means is it's a single disease-causing mutation affects multiple organs. So one gene is defective in the body in different cells, and when that gene is defective, the protein being made that's defective affects multiple organs. And usually this is a structural protein. And the best example of a disease that illustrates a principle of pleiotrophy is Marfan syndrome. And Marfan's affects 1 in 10,000, fairly common disease, and it's due to that gene that encodes for the structural protein called fibrillin. Now that protein is located, the gene is located in chromosome 15. I doubt you need to know that. You should know chromosome 15 for other reasons that we'll talk about a little bit later in this hour. But this one, I doubt you need to know that, but I have it here just to cover the bases here. And then multiple organs, and you can read this on your own, are affected. Let me ask you this. What's another example? If you're a thinker, you'll get this one. What's another example of a well-known disease that displays pleiotrophy, but it's not a structural protein? In other words, one gene is defective, multiple organs are affected. Multiple organs are affected. One gene is mutated and multiple organs are affected. Osteogenesis imperfecta could be, but on boards it'd be for another reason. And that's right, there we go. It's going too fast. Estefania, that's right. And somebody else mentioned, saw it earlier, I didn't see it, went too fast. That's right, that is cystic fibrosis. Cystic fibrosis, best thing, another great example. So if the CFTR gene is defective, remember the 3 base deletion, the cause of the disease biochemically is a 3 base deletion. It's in frame, there's no frame shift, but nevertheless, the chloride channel protein is missing a phenylalanine at position 508. As a result, that protein doesn't work, you don't transport chloride properly, salts properly, and then you end up with all the tragic situations that go on in CF. You got all the lung problems and mucus and so on. You got all that problems with the pancreas and so on, all that pancreatitis. Multiple organs are affected, all because one little defect in biochemistry. Can I say? All right, so again, that's what the word pleiotrophy means. Now the next term to review is a term called locus heterogeneity. Now without looking at this paragraph, just look away from the computer a minute and just try to picture out what does that mean before I give you the answer. Locus, because you know what locus means. We just went through that definition. Locus heterogeneity. Okay, so the way you would say this, because locus is location on a chromosome, what this must mean is that you have on either the same chromosome or on different chromosomes, you have at least 2 genes that encode for proteins, and if either gene is mutated, then it gives rise to pathology. If either locus is defective, it gives rise to pathology. That's locus heterogeneity. So, and the best example of a disease that displays locus heterogeneity is OI. And again, it's the collagen genes. Remember, we talked about that in chapter 4, the last figure in chapter 4. The collagen gene has 3 peptides. Remember, it's a tripeptide. 3 peptides wrapped around each other. But it turns out, biochemically, these 3 peptides are encoded by 2 genes. One of the genes is located on chromosome 17, and the other gene is located on chromosome 7. Again, I doubt they're going to ask you about the chromosome numbers here, but the idea is, so either gene can be mutated. Some people have a mutation in the collagen gene on chromosome 17. Other people in this world have a mutation of the collagen gene on chromosome 7. But as a result, the problem is the collagen that's going to be made will be defective. And as a result, giving rise to all the pathology. So that's what it means, locus heterogeneity. So you have multiple genes on different chromosomes, maybe even on the same chromosome, but multiple genes. If one of them is defective, then it sets off the pathology. Here, the best example is OI. Now there's a related term. Again, you should always know the name of the term and what disease is associated with it, in case they ask. Now another term is called allelic heterogeneity. Now it's shown, there's a little comment about it in your book, but don't look in your book. I'll show you where it's at in a minute. But what do you think that means? Allelic heterogeneity. If you try to memorize, you're going to get bogged down here. But what do you think that means? Allelic heterogeneity. What does that mean? Before I give you the answer. Same location for the same pathology. Not location. It's the same gene. One gene can have different mutations in it, setting off the same pathology. So here it is. This is the definition. It's shown on page 314. It's different mutation of one disease-causing locus. And depending, in other words, some people in this world may have a mutation in that gene at position 7. Other people might have a mutation, a missense mutation on codon position 19, and they're going to have the same pathology here. And that's called allelic heterogeneity. And because of where the mutation happens to be, that also gives rise to variable expression. Some people are severely affected, and some people are mildly affected. Now, there are many diseases that illustrate the principle of allelic heterogeneity. Probably the best example of this is G6PD deficiency. Now, in G6PD deficiency, you should know already from pathology that in G6PD deficiency, it's highly polymorphic. Some people are severely affected, the acute G6PD deficiency. Some people are mildly affected, chronically affected, and so on. And so they have that variable expression. And why is that? By the way, quickly, let me see your fingers do the talking here. Which chromosome is it located in? The gene for G6PD. Quickly, don't look. What chromosome, you should know. What chromosome is G6PD located on? Don't look for a number. Don't look for that keypad, because it's not a number. You look at the lower left side of your keyboard, and you see that X sitting there. And that's what you put. It's the X chromosome. Remember all that? X-linked recessive. Okay, good. See how I'm doing it? Trying to get you to recall without memorizing. Thinking, oh yeah, that's right. It's the X chromosome. All right, but now, some people in this world may have a missense mutation over here in the gene. Other people in this world have a mutation somewhere else in the gene. Other people in this, still have G6PD deficiency. Other people in this world have a mutation over there, right in the middle of the gene. Other people have a mutation nearby. And other people in this world have a missense mutation towards the 3' end of the gene. So the concept here is, depending on the person, the mutation they have given rise to the same disease called G6PD deficiency. It's a missense mutation in the same gene. And some people are severely affected. Some people are mildly affected. It all depends where the mutation is. If the mutation is towards the end of the gene, in other words, near the N terminus or near the C terminus, protein most likely is folding okay. It's going to work, maybe not great. So it might be mildly affected in variable expression. If you have a mutation in the middle of the gene, maybe affecting the active site, maybe that gives rise to severe pathology, severe hemolytic anemia, severe Heinz bodies. Depends where the mutation is. So a lot of people, a lot of diseases illustrates allelic heterogeneity. Now let me ask you another question. Now you're a thinker, you can get this one too. Not in your book, so don't look for it. What's an example of a disease in pathology that does not display allelic heterogeneity? Everybody in the world who has that disease has the same mutation. I think it's not hard. What's the name of the disease in which does not display allelic heterogeneity? Everybody has the same disease. Not Downs. No, Downs is that's not a gene defect. That's a chromosome issue. This is a gene defect here. One gene defect. Good try. At least you're trying, Leonardo. At least you're trying. At least you're participating. That's what I want you to do. That's right. And it's sickle cell anemia. Remember, everybody who has classic SS, sickle cell anemia, they have the mutation at codon 6, where the A is replaced by a T. We talked about that yesterday in molecular biology. Remember in chapter 7? Just go back to that. So the idea is that in sickle cell anemia, in the beta globin gene, at position 6, there's an A replaced by a T, and then glutamate is the codon for glutamate shifts for the codon for valine, and that is not displaying allelic heterogeneity. Now there are other kinds of hemoglobinopathies, that's true, but the classic hemoglobinopathy is not displaying allelic heterogeneity here. Okay, all right. Now the next term to talk about is a term called anticipation that I just briefly want to talk about. I want to go into the details, but what anticipation is about is it's where it has to do with those 5 diseases that are examples of the triplet expansion repeat diseases, and where the number of repeats increases in successive generations here. And as a result of the number of repeats increasing in successive generations, it sets off the disease earlier in life. Usually the disease has a delayed age of onset, but the more repeats in the gene, therefore the more earlier in life the disease sets in. So on board exams, now the example we're going to do here is one of the diseases, the triplet expansion repeat diseases, is Huntington's disease. Now what happens is, suppose you see a pedigree like this, and they show people who are affected, and they have 2 sets of numbers underneath everybody who has pathology, who has the phenotype. What do the numbers mean? In case they show you that on board exams. What that means is, on the left of the parentheses, that is the number that's the age at which the disease sets in. It has a delayed age of onset. So this male came down with the disease at age 65. His son came down with the disease at age 53. His daughter came down with the disease 55. And this female came down with the disease at age 40. So in successive generations, the disease kicks in earlier, and that's called anticipation. Now, what does the number in the parentheses refer to? In parentheses, that's referring to the number of repeats. The number of repeats. Remember, in Huntington's disease, it's the CAG repeat, and CAG encodes for the amino acid arginine. And so arginine now is one after another, after another, after another, and as a result, that is a big time problem here. So, in other words, this person in the Huntington gene had 37 CAG repeats, and then in his offspring, this person, his son, had 39 repeats of CAG in his gene. He came down with the disease earlier. His daughter has 40 repeats compared to 37. She came down with the disease at age 55. This male who had a daughter who has a disease came down to disease age 40 because they have a lot more repeats, 44 repeats. So in successive generations, the number of repeats will set off the disease earlier in life here. That's anticipation. All right, now another item here. Again, these CAGs, these are STRPs. I talked about that earlier at the end of chapter 7 in molecular biology. The STRPs are short nucleotide sequences that can be repeated multiple times. And usually it causes no pathology if those STRPs, those microsatellites, are in spacer DNA. But here, the STRPs, the microsatellites, because they're between 2 and 6 nucleotides in length, in this example here, it's 3 nucleotides. It doesn't cause the frame to shift, but the number of repeats are increasing in successive generations and sets off the disease earlier in life. Now nowadays, suppose, and by the way, in molecular biology now, is the number of repeats can be determined genetically by PCR reactions. In fact, that's discussed on page 107. That purposely skipped earlier tonight. But the idea is the number of repeats. Remember, by PCR, you're increasing the number of repeats of the exact size. So the idea is the number of repeats can be multiplied by, in a test tube, by PCR reaction, and the number of repeats likely will tell you when the disease will kick in. In other words, what I mean by this is, think of the significance. You're in medicine. Well, what's this got to do with medicine? Okay, just think of this. Suppose I am the son of this female right here. Okay, suppose that's my mother, and my mother came down with Huntington's disease at age 40 because she has 44 repeats of these CAGs. Okay, so now suppose that I'm now approaching age 35, 36. So I'm getting up to the age where if I have those repeats, I'm going to come down with the disease earlier than my mother did, because if I'm inheriting the chromosome that has the increased CAGs, so she has 44 repeats. But now if I'm inheriting the chromosome that now has a couple more repeats, maybe 46 or 47, that can be determined by PCR, that means if I'm age 35, when I get to age 36, 37, I'm going to come down with Huntington's disease. The power of the PCR reaction. So one more time, is nowadays by genetic testing, you can determine the outcome of the number of repeats. And the number of repeats will tell you basically what age the disease will kick in. The unfortunate thing is there is nothing you could do. So that's the dilemma here. So if I'm the son of that female there, when I'm approaching 35 and 36, I know I could inherit that chromosome from my mother, and man, that means that my number of repeats could be 46, 47. That means another year or two I'm going to come down with Huntington's disease, but there's nothing I could do. So do you want to determine the number of repeats? It's up to the person. Me personally, I have no idea what I would do. No idea. Maybe I would, maybe I don't know. Some people would say, well, maybe if I determine the number of repeats, in a couple of years from now, I'm going to come down with Huntington's disease, maybe I'll be a nicer person. You should be a nicer person anyway, right? And I mean, you can say, maybe I'll start smelling the roses. You should be doing that anyway. You shouldn't be worrying about things. Just, be a humanitarian, help your fellow people. And you are, because you're a physician. So the idea is, do you want to know the answer? I don't know what I would do, because I know it's going to be tragic if I have extra repeats. That is the problem here. Okay, now what's coming up next? This is going to be the last topic we're going to be covering tonight. And this topic is extremely high yield. It's another term. Let me get a drink of water here. Get ready for a lot of verbiage. All right. Now this term, a lot of students mess up on, and it's a term called imprinting. Okay, you must understand imprinting, because I'm telling you right now, Dr. Barone is going to hammer this in pathology, and I'm going to do the same thing. I'm going to hammer this home to make sure you understand it and not memorize it. Okay, so genetic imprinting. Now what this is about is a genetic imprinting. It means the differential expression of an allele depending on parental origin. What does that mean? That's just words. What does it mean? What it means is, remember, you have 20,000 genes that you got from your mother and 20,000 genes you got from your father. And on chromosomes 1 through 23, when the genes in a certain cell type, because we're diploid, when one gene is being turned on that you inherited from your mother, the same gene that you inherited from your father also will be turned on. So most of our 20,000 genes, when it's being turned on in a particular cell type, it might be heterochromatin, but if it's a euchromatin in a certain cell type, when one gene is being turned on from your mother, the same gene you inherited from your father also will be turned on. This is the genes located in chromosome 1 to 23, not the sex chromosomes. Don't worry about them. It's chromosomes 1 to 23. We are diploid for those number of genes here. Okay, so and that's the normal situation. So most of our genes, when we turn them on, they're turned on together from the mother's inheritance and from the father's inheritance. But there are rare exceptions to this, and the rare exceptions are extremely high yield. All right, now the exception. Now think normal. Think normal. We're not talking pathology yet. You'll never understand the pathology unless you know the normal biochemistry first. Now what happens is, suppose we have a father and a mother and they have a son. On the Q arm of chromosome 15, there are two locations of a pair of genes. And the way I'm going to talk about it here, I'm not going to name the name of the gene. That's going to be low yield. All I'm going to talk about here is there are two loci, two locuses, or loci. And the way I'm going to refer to this here is that there's an upper location and there's a lower location of a particular pair of genes. Upper location, lower location. Okay, it's all normal. Okay, on the Q arm of chromosome 15, all normal. But it turns out that in the upper location, without worrying about what the gene, what protein's making, don't worry about that. But what happens there is that in the upper location, the gene that inherited from the father is active. It can be turned on. But the gene that's inherited from the mother in the upper location is turned off by cytosine methylation. And that means imprinting. Imprinting. You're turning off one of the genes by imprinting. And now in the lower location, it's just the reverse. That in the lower location, the gene that's inherited from the father is turned off by cytosine methylation, never to be turned on. The gene that's inherited from the mother is active. And again, this is normal and it's called imprinting. Now let me stress as much as I can. Get it through your mind that imprinting is normal. A lot of students think that imprinting is the pathology situation. It's not. Imprinting is the normal situation. Get it through your mind. Write it over and over again until you master the concept that imprinting here is normal. It's unusual, yes. It's rare, yes. But it's normal situation. Because these two genes are not in the upper location, lower location. In the upper location, both genes are not turned on. In the lower location, both genes are not turned on at the same time. Because one of the two genes in either location is imprinted. It is the normal situation. All right, let me ask you this. So far, I'm taking this extra slow. Does that make sense so far? Let me see. I want you to know this. Am I clear? Am I mumbling? Okay. I see all the yeses there. Okay. Okay. Makes sense. Okay. Everybody's okay. Okay. Syed, no. Okay. Listen. Okay. Here it comes. Let me repeat because this is so important. Most of our 20,000 genes on our chromosomes 1 to 23. You have two chromosomes, one inherited from the mother, one inherited from the father. The genes that when they're being turned on, that inherited from the father, when they are being turned on, are also being turned on in the mother. We are diploid, and both genes are making RNA. Both genes are making protein. Like what genes? 20,000 genes, you name it. And most of them are being turned on at the same time. The genes you got from your mother and your father. The exception to this is in the Q arm of chromosome 15. There are a pair of genes. There's an upper location. There's a lower location of the genes. Don't worry about what the name of the genes is. Make it simple. Don't worry about the genes, but there's an upper location, and it just so happens the gene that's inherited from the father in the upper location is active. It's made into RNA, made into protein. The gene that's inherited from the mother is never turned on because it's imprinted. It's cytosine methylated. It's imprinted. So only one of the two copies is turned on. Normal, normal. Capital N, capital O, capital R, capital M, capital A, capital L. Normal situation is imprinting. Write it down 1,000 times. In the lower location, it's just the opposite. The gene that comes from the father in the lower location, what it encodes, not important. But the gene in the lower location is printed by cytosine methylation, never to be turned on for the rest of your life. Whereas the gene you inherited from your mother is always on chromosome 15. See, yeah, does that make sense? Please say it makes sense. Okay, good. I want you to know this. Sometimes I'm kidding around. By the way, never take anything personal. If I make a comment, don't think, oh man, he's criticizing me. Don't do that. We're among friends. I'm just trying to joke around sometimes, and sometimes people get offended by a particular comment. Get a life. I'm not criticizing anybody. If you don't know, I'll joke around. Don't get upset and email Kaplan Medical and say this guy Turco is a jerk. Get rid of him. You know, this kind of stuff. Don't. Get a life. If I'm joking around and I say something you think is offensive, although it's not, I'm just joking around. Get a life. Don't get bogged down here. All right. Okay, so that's the normal situation, and you'll never understand the pathology unless you understand the normal situation. Now, what can happen? Here comes the pathology that Dr. Barone will talk about. And the sooner you master this tonight and tomorrow, by the time, and I mean mastering, be able to verbalize what's going on, explaining what goes on. When Dr. Barone is here, you'll know exactly what he's talking about. It'll make sense what he's talking about because you know the biochemistry first. Now, what happens is in the upper location only, the gene that comes from the father is deleted. For some reason, the gene got deleted. Remember, sequences of codons can be deleted. Remember CF, cystic fibrosis, you can have deletion of a codon. You can have a deletion of a nucleotide. Remember all that? And here, a whole gene is deleted in the upper location of the chromosome coming from your father. And in the upper location, the gene that comes from the mother is still present, but it's imprinted. It's never turned on. The lower location, nothing wrong. Nothing wrong with the lower location, it's fine. But in the upper location, the chromosome that comes from the father has a deleted gene. The chromosome that comes from the mother has the imprinted gene. Now, what does that mean? Now, think about it. How does this fit into biochemistry? What that means is, in the upper location, it's making no functional enzyme. You don't have to worry about this. It's not important. But the gene encodes for a protein involved in the spliceosome. Don't worry about it. I'm not going to ask that. In fact, it's still being researched. But the idea is a gene is deleted and the other gene is imprinted. There's no functional gene, no functional enzyme, and going to give rise to pathology. And what's the name of the pathology? Everybody knows it's Prader-Willi. So Prader-Willi, now Dr. Barone will talk about all the pathologies, all the issues of the characteristic hallmark is insatiable eating, always hungry. It can have some mental retardation, webbed fingers, and all these type of things. But the main issue is insatiable eating, is Prader-Willi. All because in the upper location, there is no functional gene. Now, if on the boards they ask you, what's the molecular basis of the pathology in Prader-Willi? Option A will say imprinting. Option B will say deletion of a gene. Most medical students are going to put option A, imprinting. Imprinting, get it through your mind, is normal. It's normal. The gene coming from the mother is normal, but the gene coming from the father is deleted. That's what's causing the pathology. Please understand that. Understand that. Master it. Don't go memorizing this. That's what's causing Prader-Willi, and you say in the upper location of the Q chromosome 15, there is a gene that's coming from the father that's deleted. The gene that comes from the mother is imprinted. It's normally imprinted. And so therefore, there's no functional gene, no functional RNA, no functional enzyme, giving rise to pathology. Now, if that happens in the upper location, you might imagine this is going to happen in the lower location, right? And the same thing can happen in the lower location. So in the lower location, now let's focus, let me highlight it in the green box, the lower location. The upper location is normal, nothing wrong. But in a lower location, the gene that comes from the father is imprinted. That's normal. There it is, normally imprinted, cytosine methylated, turned off normally. The gene that comes from the mother's side is deleted. The gene is missing. So what that means is in the lower location, there is no functional gene. And therefore, no functional RNA being made, and the RNA is then made into protein, and you're going to have a problem in the cell. Now, you don't have to know this either, but the biochemical defect is known. It's due to the proteasome, the ubiquitin proteasome stuff we talked about earlier. That's the defect in Angelman syndrome. So Angelman syndrome has a defect, a dysfunctional proteasome. And the reason is because in the lower location, one of the genes is deleted, giving rise to Angelman. Now, Angelman syndrome, again, when Dr. Barone is here, he'll talk about the pathology. And let me see the answer here. There we go, man. You beat me to the punch, Natalie. There we go. I was going to ask, what is the hallmark? And that's the happy puppet syndrome. where those infants are always laughing, or children are always laughing, and they always have a kind heart. Always, if you're ever depressed about something called the USMLEs or whatever, you're getting ready to take it, and oh man, I'm depressed. Go see a child with Angelman. They'll warm your hearts because they have such a tragic disease, yet they're happy. The happy puppet syndrome. Now again, if on board exams they ask you, what is the molecular basis of Angelman syndrome? You answer by saying, well, if option A, they might say option A is imprinting of a gene. Option B, they may say deletion of a gene. And the answer is what's causing the pathology is deletion. Please understand. Okay, everybody with me there? Okay, again, I took it extra slow because I want you to know. Okay, and again, Dr. Barone is going to be talking about this, and the sooner the better. Now, one more item, one more figure, then we're done. But somewhere else in one of the other chapters, there's another term that I want to cover now, since we're talking about terms and diseases associated with that term. So I want to cover it now, even though it's talked about later on. And you'll see why I want to cover it now. And this term, this last figure, so it's just use a blank figure, it's called uniparental disomy. What does the words mean? Look at the words. What do they mean to you? Uniparental disomy. What does that mean? I mean, those are words. What does it mean? What does it mean? That's right. That means disomy means two chromosomes, not all the chromosomes, but two chromosomes. You have two copies coming from the same parent. So two copies of the same chromosome being derived from the same parent. Not all the other chromosomes, but one of the chromosomes is now you have two copies of it coming from the same parent. That's what it means. Uniparental, one parent, two chromosomes. Okay. All right. So now, what's this got to do with pathology? Okay, that's a term in genetics. Well, what's this got to do with anything? Okay, here it comes. Here it comes. You'll see. Now, what happens there is, let's talk about the same couple here. Here's a male and a female, and they have a son. On the Q arm of chromosome 15, there are these two loci, two locations of two genes. Upper location, the gene that comes from the father is active. The gene that comes from the mother is inactive by imprinting. And lower location, it's just reverse. Now, what can happen? What can happen in meiosis? Don't worry about the details. But what can happen is the son might inherit both chromosomes coming from one parent. And one of the two terms is called paternal disomy. That means the lack of the maternally derived chromosome. In other words, in this diagram, suppose this offspring inherits two chromosome 15s coming from the father, not the mother. Despite this, just the fact in meiosis and so on, two chromosomes 15 coming from the father is going into one zygote. And so then the zygote now inherits two chromosome 15s coming from the father and no chromosome 15 coming from the mother. Here it comes. From pathology, what disease does that give rise to? What disease in pathology that Dr. Barone may talk about? He should talk about. I know he will. What disease is this? Think. Don't go memorizing, you'll get it. No, not that. You're overcomplicating. No, there's no trisomy 18. There's no this is chromosome 15. Why are you talking about chromosome 18? This is not Edwards syndrome. This is chromosome 15. What's it? There we go. We're close. What does that mean? That means in the upper location, there is a functional gene. If in here are two chromosome 15s coming from the father, then here it is. Let me draw it out for you. Notice here in the offspring, in the upper location, there are two genes that are now active. That's not the problem. The problem is the lower location. In the lower location, there is no functional gene. And therefore, you're going to end up with problems with the proteasome and all this type of stuff. And what does that give rise to? That gives rise to Angelman syndrome. Now, Natalie, when you said Prader-Willi, you were close. You were close, so I'll give you a lot of credit for that. But this is Angelman. Why? Because at the lower location, there's no functional gene. And the reason is because both genes are imprinted. No functional gene. Okay? All right? Does that make sense? Makes sense? Okay? Makes sense? Okay. Now, here it comes. One more thing, then we're done. Perfect timing. Now, if there's something called paternal disomy, you can imagine there's something called maternal disomy, where there's a lack of the paternally derived chromosome. See this? See the situation over here? Suppose an offspring inherits 2 copies of chromosome 15 coming from the mother. So as a result, it's going to look like this. So that offspring's chromosome 15 will look just like this. In the upper location, there is no functional gene. In the lower location, there is 2 copies of the gene that are being expressed. That's not the problem. The problem is the upper location is making no functional protein. As a result, the spliceosomes don't work very well and so on. Don't worry about the details there. But the idea is, what's the name of the pathology that gives rise? Now, Natalie, now I need you. What disease, what pathology does that give rise to? That's right. Yeah, no. Bingo, bingo. That's right. Prader-Willi syndrome. There it is. So that's why I'm covering it now instead of later on, because we just talked about the most common cause of Prader-Willi and Angelman is due to a gene deletion from the previous page, the most common kind of Prader-Willi and Angelman. But the rare examples is due to uniparental disomy. So even though it's rare, the National Board asks about it because they want you to understand the molecular principle of what's going on here. So even though in your lifetime you may not see a patient who has Prader-Willi due to maternal disomy, but you might, but they expect you to know what's happening. Don't worry about the details. It's the concepts here. All right? Okay, everybody, so we're done for the day. Perfect timing, 10 o'clock, perfect timing. So what we're going to do is we're done with chapter 1. So tomorrow we'll come in right in the first hour. There'll be several vignettes, clinical vignettes that are going to be coming your way. Actually, we're going to do the vignettes at the end of chapter 2. Let me say something about chapter 2, by the way. Population genetics, and that's that Prader-Willi, that's that Hardy-Weinberg. In my opinion, don't read ahead. You're going to get bogged down. This population genetics can be very hairy if you try to understand it on your own. So if you come tomorrow, I'll try to explain it as best as I can, but you must know the Hardy-Weinberg. That's the only thing. This is a fairly short chapter, even though there's other things in the chapter, but we're only going to focus in the concept of population of the Hardy-Weinberg. So it takes about 20 minutes or so to go through that the Hardy-Weinberg, but I'll tell you what you need to know. So in my opinion, don't read ahead. You might get bogged down and then hate me for it. Because I want to tell you, don't worry about that. So I'll tell you what you need to know. Okay, so then after we do chapter 2, then there'll be a series of 3 vignettes that we're going to, vignettes based on chapters 1 and 2. Now again, after I'm done right now, I'm going to post on Facebook 3 vignettes. And again, there'll be question 9, wait, not with question number. The number of the questions. But in any case, questions 10, 11, and 12, they'll be posted. Again, explain your answers. That's to your benefit. Explain your answers. And then tomorrow afternoon, before we come on board, I'll provide the answer. Okay, everybody, thank you so much for attending. Thank you so much for bearing with some very difficult concepts here. But you guys are doing great. I know it's not easy, but you guys are doing great. And thank Dr. Donald, Dr. Seda, and Roy and Steve for all their help. They've been fantastic here, and appreciate all you guys for what you do. Thank you so much, and I'll see you tomorrow.